Do we have a mic?
Yeah, we've got the power if you prefer, but that's probably better. Yeah, it's on mute. Flick up to. Let me set speaker. Yeah, so the screen should be on there.
Hello, is this on? I think it is on. Hello and welcome everyone. Welcome to our Bragg Exchange 2024. It's my pleasure to open this. I'm Edmund Linfield, I'm Director of the Bragg Centre, and it's really great to see you here, and thank you for all those who braved wind, rain and everything else to come to rather damp Leeds. But I have to say, it's probably damp anywhere in the UK at the moment, and probably the Northern Hemisphere, so I'm not going to take any responsibility for the rain in Leeds. It's really great to see everyone here for our annual materials meeting. Really looking forward to seeing it all, and I've already been to the refectory where we're doing the posters and had a sneak look, and there really are some excellent posters down there. I'm not going to say very much because I'm going to introduce our Deputy Vice-Chancellor in a second, but I think before we start, and the fact you're all here with badges and everything else, I want to say at the start a big thank you to Andy here, Lucy and Helen, who've really put all of this together. So I'm just introducing, but the fact you're here is nothing to do with me whatsoever. So without saying any more, I'm going to introduce Professor Nick Plant, who's our Deputy Vice-Chancellor for Research and Innovation, if I've got his title right, who's going to say a few words at the start to welcome you. Nick.
Thank you very much, Edmund. It's a real pleasure to be back at the Bragg Exchange. So last year we had over 200 delegates. The public broadcast reached 600 people and growing. This year we have 300 delegates registered. Who knows how many people will watch the public exchange, and I know Andy's been working on some new and exciting things, including a clean room VR experience. If you haven't been able to see the clean room yet, this is your chance to do it. And trust me, it's a lot easier than putting on the blue suit and actually going into the room itself. So the University of Leeds' strategic vision for research and innovation says that we'll be at the centre of a global research and innovation community that will deliver transformational, fundamental and challenge-led research to advance knowledge and tackle major issues. The University of Leeds, I believe, benefits from excellent facilities, a strong research culture, and most importantly, excellent people. I genuinely believe that when you put those three things together, there's not a lot we can't do if we put our minds to it. So one of the challenges I would give to you as a community is put your minds to it and decide what it is we want to achieve, and then go out there and do it. The Bragg Centre is a great example of everything I want to achieve through the university strategic vision. It has fantastic staff and students. It has great collaborators and partners out in communities. A really strong research culture, I think one of the best across the university, and a good environment with fantastic facilities. Because of that, Bragg does deliver both fundamental and applied research that truly is world-leading. Now is not the time to rest on your laurels, however. I'm going to again challenge you. I think you can do more. I think as a community you are only just starting, and there is much more to come. The Bragg Exchange is one of the centerpieces for what Bragg does, and it epitomises the vision to bring this vibrant and innovative materials research community together to deliver the greatest impact across disciplinary boundaries. The fact that you can bring so many different disciplines in one room really shows why Bragg as a centre, and the Bragg Exchange in particular, is special. It genuinely helps us fulfil what we want to do as a university. So over the last year, I think the Bragg has really focused on research culture and environment, and in particular championing its research technical professionals. I think that the activity of this group is what powers the research that we do. They help the design, they help the delivery, the interpretation, and they help working with our external partners to really spread the word of why the Bragg Centre is genuinely world leading. I and the university really welcomes this focus. It's provided a really good forum for me to come out and listen to how it really works on the ground, and I genuinely value that honest feedback. I do sometimes come out feeling slightly bruised, but that's always a good thing. It's a good thing to be told where we can improve. And I think this willingness to champion has really helped drive some of the new policies that we see coming out across the university. So, for example, the work that we've done to realise a 50% reduction in our fixed-term contracts. The fair attributions policy that will be going through to council next week that will ensure that we recognise on our outputs all of those individuals who have contributed to the research and therefore warrant either an acknowledgement or a name on the author list. Finally, I think that the centre has really worked hard to support the skills pipeline, working on a nationally acclaimed schools outreach programme, undergraduate research internships, and funding PhD studentships. This year, I know one of the focuses is on advanced skills training programmes to help meet the UK's semiconductor industry needs. And again, I think we can be really proud that today we have generated some of the research leaders. And if you come back in 5 years' time or 10 years' time, if we're all still here, we will be saying the same thing. We will put out a new generation of research leaders, the people who will go out into the world and shape how we think about the future. That, to me, is why the Bragg Centre, and in particular events like the Bragg Exchange, where we come together and we talk about our ideas, is absolutely critical. I really look forward to seeing the Bragg Centre continue to grow over the coming year and working with you to help realise that vision. Have an absolutely wonderful conference, and I look forward to seeing some parts of it as I stay around. Thank you very much indeed.
Thank you very much indeed, Nick. And as you said, working together, working across disciplines, working across different universities and institutes in the country is very much what we want to do. So the second part of this introduction is really over to Andy, and Andy is going to tell us what we need to do because he is the one in charge of everything. Okay, Andy,
thank you very much.
With that introduction.
Thank you very much, Edmund, and I just want to second that it's wonderful to see so many people in the room. The conference has continued to grow year on year. As you'll note, we've spread to 2 days now, just to make things, manage to fit everything in, and it's really wonderful that you've all given up the time to be here as part of that. So some practicalities, fire alarms. We are actually, the fire test is actually tomorrow morning. It should be before we actually arrive back in the building for tomorrow morning. So if you hear any alarms whatsoever, it is actually a real alarm. Fire exits, very convenient in this room, are at the bottom front here. And of course, if we're out in the foyer, then it's straight out of either of the main doors. The congregation point is on the grassed area outside the main entrance, which is that side of the building, the other side behind you. In terms of first aid, we have plenty of first aiders in the room. In particular, we have Helen and Lucy, who are part of the Bragg team, who are first aiders and will be able to assist you. Do let any of the staff in helping the event know if there's any emergencies. Nearest toilets, just down this corridor and behind you. Basically where the coffee was early this morning. They're underneath the lecture theatre that you're sat in at the moment. And then for lunch and posters and exhibition, we're actually going to move over to the refectory. So we're going to leave by this exit and head 5 minutes down the road to the refectory to spend about an hour with posters, lunch. We'll then come back here for this afternoon's talks, and then the reception for drinks and VR experience and posters will be there in this evening again. So there is a little bit of back and forth, but we will help guide you the entire way. Unfortunately, we didn't bring brollies for everybody though, so note for next year, branded brollies. Just to remind you, hopefully you're all aware, but this is a being live broadcast as part of the Bragg Centre's mission to widen access to the science that we do. Then we have a live stream that's going out to YouTube for public to engage with. And I know that there are many members of industry who are signed up to watch this as well as some schools and people involved in our public engagement networks as well. So hopefully we should get a really good audience, and then it will be available on demand afterwards for people to pick up as well. If you're uncomfortable being on camera, I would note that it's mainly focused on the speakers, so maybe sit further back towards the back of the room and you won't be in the broadcast. So you may notice that we haven't got any paper agendas printed out. This is part of a sustainability mission that we're trying to achieve across the university widely, but also champion in Bragg. The agenda is available digitally. There is a QR code there if anyone wants to scan it quickly. However, if you have a look on the back of your name badge, you might have to move your little poster voting slip slightly out the way, but there is a QR code conveniently there for you to scan, which will give you access to the digital guide, which has all the information, including the full agenda in it. And I noticed loads of people getting their phones out now. Great. So it's available for you at any point, so it's on the back of your badge. Final thing then, we also have a poster voting slip, which is also tucked conveniently in the back of your name badge. So this, when we go over for the poster sessions, there are 3 poster sessions, one at lunchtime today and tomorrow, and then this evening as well for the drinks reception. Poster voting is on a public vote, so the highest votes win. Everyone has a single slip, so everyone can cast one vote, and you simply write the number of the poster that you would like to vote for in the box and drop it in the conveniently placed boxes around the room. It's very, very simple. And you have until tomorrow at the end of the final poster session to do so. And we will then count and award the posters. Finally, I'd like to note that this year we have some sponsorship for the exchange. Very grateful to Quantum Design for sponsoring the exchange, but also in particular, the Royal Society for Chemistry, who have sponsored the poster prizes as well as offering some money to help support the event itself. So alongside the poster prizes, of which there are going to be 3, the first prize and then 2 runner-up prizes, alongside some Amazon vouchers that the Centre and the RSC are funding, you will also get 1 year's free RSC membership as well with that prize. So make sure that you're all presenting your posters to the best that you possibly can. It's going to be you want to gain as many votes as possible. And with that, then, I'm going to hand over to our first chair, Professor Rick Drummond-Bridson, who is the theme lead for the analytical science theme in Bragg, who is going to chair our first session.
So this first session is focused on analytical science, and we've got a guest external speaker, who's Professor Melanie Bailey from the Surrey Iron Beam Centre in the Department of Chemistry at the University of Surrey. And she's also an EPSRC fellowship holder at the moment. She obtained a BSc in physics in 2001 from Manchester and a PhD in electrical engineering from Surrey in 2005. And she worked as the Ion Beam Analysis Liaison Fellow for the Ion Beam Centre between 2007 and 2011. And during this time, she began working with various law enforcement agencies to develop ion beam analysis methods in forensics, and has worked as a consultant of the International Atomic Energy Agency to set up a research program on nuclear techniques in forensic science. She was appointed a lecturer in chemistry at Surrey in 2011 and subsequently gained a chair. She researches analytical chemistry and accelerator physics, and as well as ion beam techniques, she also runs a mass spectrometry facility. Her research interests centre around the analysis of small sample volumes using mass spectrometry and ion beam analysis. And this spans samples from semiconductors, drugs, other biomarkers, through to the analysis of single cells, as we'll hopefully hear about. So it's my great pleasure to welcome Melanie to the podium. Thank you.
Okay, hello everybody. Can you hear me okay? Yeah, nobody's shaking their head, so I'm guessing yes. All right, well, thank you so much to the organisers for the opportunity to come and talk here today. You have to forgive me, I get very travel sick on trains, and I had a 5-hour journey to get up here, and I took some travel sick medication, which has made me very woozy. So this is the closest I've ever been to being drunk while giving a talk. So we could have some interesting discussions. What I wanted to come and talk to you about is our new and very exciting Wolfson Analytical Centre, which I'm hoping will operate in a similar manner to the amazing Bragg Centre that you have here, but offers some complementary capabilities. And so what I'm really trying to do here is to advertise to you some of the perhaps more unusual approaches that we have at the University of Surrey, which you might be able to make use of in your own research. So the funding's recently been awarded from the Wolfson Foundation to sort of concatenate all our research interests in bulk biomarker analysis, imaging, mass spectrometry, and then kind of weird and wonderful ways of testing patients in clinical situations. And so the idea is that we're going to come together to build all these analysis and sampling protocols that are faster, smarter, greener and smaller. So it's a collaboration of a number of different academics from across the university, spanning physics, electronic engineering, chemistry, and then several different disciplines within biosciences as well. So we're getting a new building, which should be ready in 2024. And the idea is that we have all these different pieces of instrumentation and capability which are currently scattered all across our university campus. And I've kind of plotted them as a function of their lateral resolution and available information. So, we have some of the perhaps more routine mass spectrometry instrumentation for bulk, lipidomics, metabolomics, proteomics, but we also have systems for that are sort of not available anywhere else internationally, which I'm going to talk about at our National Ion Beam Centre. So we can get a great deal of information, biological information, at all sorts of different scales of resolution. So I'm going to start off by talking to you about the sort of unique analysis capability that we have at the UK National Ion Beam Centre. So the Ion Beam Centre is an EPSRC funded facility. So if you are applying for time on EPSRC grants, we can provide you access without incurring any costs to your grant. The costs are already included within our facility grant. So at the UK Ion Beam Centre we have, or at Surrey, we have various different accelerators which we use both to modify but also to analyse the composition of materials. And so what I'm going to talk about here is mainly on the analysis. So we have an accelerator that we can use for various different things, high-resolution depth profiling of elements, and we can also use it for high spatial resolution mapping. So we can go to submicron dimension. So here you can see what happens when the ion beam hits the target. So what the accelerator does is it fires an ion beam, so maybe protons, at a sample. By the time it hits the sample, it's going at about 10% of the speed of light. And then various things happen when it hits the target. So you get light, you get particles emitted, X-rays emitted, gamma rays. And what we can do is analyze all of these signals simultaneously and self-consistently to build up a very comprehensive picture of the elemental composition of our sample and its variation laterally and as a function of depth. Now, because ion beam techniques are not mass spectrometry techniques, the matrix interferences are very limited. So it means that our capacity to, or I guess the images are less artifact prone than techniques that use mass spectrometry. So here you can see some images of, this is just one cell, and you can see thallium, a radionuclide that's been delivered to the cells. And actually, it's not shown here, but the techniques have sufficient spatial resolution so that you can see whether or not a drug is delivered to the nucleus or the cytoplasm of the cell. So I have an EPSRC fellowship that's looking at multimodal imaging using ion beams and mass spectrometry imaging. So what we're trying to do is we're trying to develop a suite of techniques that can be used to look at elemental and molecular biomarkers at subcellular resolution. So we're really trying to get inside the cell and really try to find out what's going on at an unprecedented scale of detail. Now that all sounds reasonably straightforward. Of course, you could perform the two imaging modalities on different samples, but it's very difficult to reproduce a single cell because they're so heterogeneous on two different samples. So what we're trying to do is develop a suite of techniques that can be used on the same section of tissues or the same sample of cell culture. So I guess the other thing to say here is that elemental imaging is being provided by our Ion Beam Centre, and then mass spectrometry imaging, which looks for lipids, metabolites, perhaps even proteins, is being provided through MPL, which are a sort of national centre of excellence for mass spectrometry imaging. So the sorts of things that this approach can give you is perhaps if you're delivering metal-containing nanoparticles to tissue, you can start to understand their interaction with the host lipids and metabolites, perhaps understand the sort of impact on the local metabolome or lipidome of them being there. So sort of along this vein, we've been looking at nickel nanoparticles delivered to skin, and their impact on the skin tissue. But then there's also elemental markers from natural accumulation of metals in biological systems. So for example, in tuberculosis, we've done some work with researchers in the US where we've looked at the distribution of different elements in a lesion called a granuloma, which is caused when an organism gets tuberculosis. And you can see a buildup of iron. And iron is very important in tuberculosis, but one of the problems with iron is that it also causes oxidation of lipid membranes. And so what we're trying to do is develop techniques that we can use to co-locate iron and lipids to try to understand that interchange between those elemental and molecular markers. And so here, if you want to go and have a look, you can see some of the papers that we've published. And these are our sort of co-locations of different elements and and molecular signatures that we've been able to produce. Now, conventional mass spectrometry imaging techniques, so you might have heard of MALDI, matrix-assisted laser desorption ionization, or DESI. These are two commercially available mass spectrometry imaging techniques, but they work at quite large scales of resolution compared to ion beam analysis technique. With an ion beam analysis technique that looks for elements, we can easily get submicron, well, not that easily, but we can get submicron resolution. With mass spectrometry imaging, you're typically looking at pixel sizes down to about 5 microns if you're lucky. You get very limited information as well at those kind of pixel sizes. So we've actually been looking at secondary ion mass spectrometry as our mass spectrometry imaging technique, because with secondary ion mass spectrometry, you can actually start to meet the spatial resolution of the ion beam analytical techniques. And we found that the two processes are compatible, and there's recently been a development in secondary ion mass spectrometry. So I don't know if anybody here, is anybody here a user of SIMS? Yes? A couple? Yeah, okay. So with SIMS, one of the problems that you have is fragmentation of molecules, right? So you get so much fragmentation, it can be very difficult to do your peak assignment. And in the last decade, there's been this amazing advancement in SIMS, the use of water cluster sources, which are much gentler, so you get amazing coverage of biomolecules compared to conventional SIMS, but with really good spatial resolution. So you can see what you've got and also where it is, importantly. And what we found is that we can couple together ion beam analysis and SIMS, perform them on the same sample so that we can really at 1 micron, 2 micron spatial resolution co-locate things like iron and lipids or nickel and lipids and metabolites. So this is great, but we're unable to do this on the same system, which can cause some problems. So we have to take the sample out of the vacuum chamber in order to analyze it, and then it can be quite difficult to relocate exactly the same position of the sample, and also the sample has gone in and out of vacuum chamber, so it kind of changes its configuration, and it makes the registration of these two images really, really difficult. And so recently we applied for some funding from the EPSRC to build a system on which we could do both KEV and MEV ion beam measurements. So now in the same system, using our accelerator, we're going to be able to do molecular and elemental mapping. And so the idea is that we should be able to do 1 micron resolution elemental and molecular mapping on the same sample with this system. And this also has some other advantages. So you can also use both types of ion beam for depth profiling. And so we'll be able to do multimodal depth profiling of organic and inorganics at hopefully unprecedented spatial resolution and coverage. We have to get the instrument before we can prove anything. And then the other thing is that there are lots of ion beam sensors internationally with MEV beams that actually use the MEV beam to release molecules from the surface of the sample. And the MEV beam has some advantages compared to the lower energy beams because they can release much larger molecular species. And so again, this will give us an optimized mass spectrometry system in which we'll be able to fully explore this. Up until now, people have made their own mass spectrometers to be able to sort of take mass spectra from samples using MEV beams. And of course, that's completely suboptimal when you think about how much development has gone into mass spectrometry in the past few decades. So that's sort of multimodal. So I guess the message to take away there is we can do multimodal mapping. We have different ways of, different methods for registering images and looking at co-location of elemental and molecular markers. And so if you're interested in that, do get in touch with us. The next thing to talk about is another unique facility that we have in the UK. And this is for live cell mass spectrometry omics with spatial information. So this is a BBSRC-funded facility, and like the rest of the centre, the aim is to provide measurements for the UK biosciences community. So in this case, the target is BBSRC remit research. You don't necessarily have to have a BBSRC grant, but you do have to be working in a BBSRC remit area to get access. So what we actually have is a system that operates, it's a confocal microscope. You can take beautiful live cell images, so your cells are living at the point of sampling, and you can choose a particular point in time or space to sample your cells. So, you may infect cells with a pathogen or a drug or apply some kind of biological stimulus, and then choose a particular cell at a particular time to perform sampling. And so what the tool can do is it's a robot with a capillary, and you can pick up a cell into the capillary, and that cell is captured then for downstream analysis. And it only captures one cell. It doesn't capture the neighbours, so you can go back and, if you want, sample the neighbours. So imagine that you had an infected cell and you wanted to try to explore the interaction between that infected cell and its neighbours, or you wanted to you'd hit a cell with some kind of radiation or a drug, and you wanted to, again, explore spatial effects. This tool will allow you to do this. The other thing that we can do is subcellular sampling. So here you can see these are nuclei within cells that have been stained using a live cell imaging dye. And here you can see a nucleus being extracted from a cell. So here you can see this is the before picture, and this is the after picture. So it's just literally being hoovered up into a capillary. Here you can see we've actually managed to pick out, this is a cell that's infected with BCG, which is like a safe version of tuberculosis. So the BCG is fluorescently labelled, and you can see we're actually able to extract that BCG and perhaps some of the surroundings from that cell, leaving behind the rest of the cell. This is showing, these are stress granules that form within cells, and they're of significant interest to virologists because these are the factories where the viruses replicate. And so virologists are very interested to find out what they're made of. But if you analyse the whole cell, of course, you see what the stress granule is made of, but also everything else in the cell. And so what we've been doing is selectively extracting these stress granules into capillaries in order to analyse them. And then this is showing you, perhaps a little less convincing, but sampling of, so these are stains. The stain is a mitochondria, and you can see here that we've been able to select a region of the cell that contains or is rich in mitochondria. So basically, this is a platform technique for being able to pick bits of cells, all whole cells, under a microscope while they're living, collect them into capillaries, and then you've got your sample captured into capillary, and there's various things that you can do. So you could perform analysis to look for elements. You could perform mass spectrometry analysis to look for lipids, proteins, transcripts, genes, metabolites. It's basically a platform way of picking out bits of sample. And so the idea is that some of the analysis is being performed at our facility, but there's also the option to take your little bits of cells home and analyse them in your own lab as well. So some of the things that we've done so far is we've developed a method for single-cell lipidomics, and we get around, depends what kind of quality of information you want, we see around 400 lipid peaks per single cell. We can get very high quality information from a smaller number of peaks, so maybe 180 or so. Depends on the certainty with which you need your identification of your lipid. And anyway, what we've been able to do is see the difference between cells that have been treated with hydrogen peroxide. So here you can see some control cells and some treated cells. They separate nicely using principal component analysis. And we can see the different lipids that are changing as a consequence of that treatment. We're also able to use single-cell lipidomics to see the difference between cells that have and have not been treated with chemotherapeutic drugs. And we can also see responses to radiation. So if you irradiate cells through a pinhole, you can see differences between the cells that were irradiated and the bystanding cells, which is exciting. So, as I said, this is the seismic facility. It's available for people from all over the UK, and if you want to use it and you're working in an area that's relevant to BBSRC, we'd be very happy to come and talk to you and see if we can help you with your research. So the last thing I wanted to talk about was a sort of a niche part of our biomarker analysis, which is faster techniques. And so this is all about non-invasive patient testing through fingerprint and sebum sampling. So this work actually started in forensics. I started working with the police to use ion beam imaging techniques and see what we could do with them in forensics. And I guess one of the first things that we demonstrated was that you can use mass spectrometry imaging to reveal missing information from fingerprints. So I still have a PhD student. She's a part-time PhD student. So it's been a very long project. And she works for Thames Valley Police as her day job in the fingerprint department. And she uses, at the University of Surrey, mass spectrometry imaging. So in this case, we're using time of flight secondary ion mass spectrometry, or TOF-SIMS. We're using that to visualize the missing information in fingerprints that the police can't see. So this is a fingerprint that's been deposited on a surface, developed using cyanoacrylate, and you can see that it's only a partial development. And if you go to the one of the sort of areas where there's no visible print, it's very easy to actually visualize that missing information using mass spec imaging. And so what my PhD student has been doing is sort of seeing how applicable that is to different types of surfaces, different types of developer, different donors' prints. And excitingly, we've been able to get the SIMS technique into the fingerprint visualization manual, which is the manual that the police use for as a guide for fingerprint visualization. So moving on from that, it was suggested by colleagues and collaborators working at the Home Office that there's a lot more that you can get out of a fingerprint than just the 2D ridge structure information. And when we're doing mass spectrometry imaging, of course, we're detecting thousands of different molecules within a fingerprint. So can we use that to gain more intelligence about the person that deposited the fingerprint? Because a fingerprint is a combination of sebaceous fluids touching your face, and eccrine sweat, and also substances that a donor has touched. So can we use a fingerprint to determine the age or the sex or even drug use of an offender? And so we've, over the past years, targeted our research towards looking at drug use from a fingerprint and what it really means to detect a drug in a fingerprint sample. Now there's a big sort of economic driver for doing this. It's not only forensics, but also drug testing. So could you use the fingerprint as a really easy way to test somebody for drugs? It's really fast. We spent 6 months in hospitals taking samples from COVID patients during the pandemic. And we collected fingerprints, we collected sebum, blood, saliva, and the sebum and the fingerprints were so easy to collect, and it was an absolute nightmare to collect the other biofluids from patients. So there's a real driver, both from the patients but also from the clinicians, to try to develop tests that are non-invasive. And so from a drug testing perspective, you've also got the fact that the identity of the donor is embedded in the sample. So you can see, you've got a sort of traceability thing there. You can see who deposited the sample. So we've looked at cocaine in fingerprints. There's lots of publications that say that you can see cocaine in fingerprints. And we've been able to, we've explored the difference between people who have handled illicit drugs and people who have taken them. And so here you can see with mass spectrometry imaging, it's very easy to tell the difference between people who've handled drugs and people who've ingested them. When you ingest a drug, you have a much more consistent image of the drug and the metabolite in the fingerprint. So we've also extended that to looking at therapeutic drugs now with different NHS trusts. And we've used a fingerprint test as a tool to see if a patient is taking their medication. So for example, we've done this with antipsychotic drugs. The test works really well. We can see the difference between people who have touched and taken the drug. We are also looking at antibiotics. And again, the test works really well for antibiotics as well. I guess the problem really comes into how you quantify the level of drug present in a fingerprint, which is a little more tricky and something that we've been working on with Groningen Medical Centre. So I'm going to skip to the conclusions because I'm aware of the fact that we're running out of time. Really, I'd just like to say thank you very much for listening. I hope that it's given you sort of an overview of some of the very varied things that we're doing within our centre at Surrey. And if there is anything that sort of rings a bell, then do come and chat. If there's anything that you think you could make use of, then please come and use us. And thank you very much for listening.
Thank you. Thanks for a great talk, very interesting. When I was holding my fingers up, it wasn't to get fingerprinted, by the way. Any questions for Melanie about either the maths? Jim?
Can you hear me? So the SIMS mapping is quite interesting, and the fingerprint example brings up a question about the scale. I'm surprised in a way that a technique that has that length scale of resolution can be used on an object that has such a large size. So what are the limitations in terms of the area that you can image? And I realize that depends on what you choose for resolution, but
Yeah.
you know, for a fingerprint, how long would it take, or do you just look at a tiny little region?
Yeah, so that's a really good point. I think it depends on, so as you say, you can choose the area. You can do a whole fingerprint with SIMS. It takes about 3 hours.
That's it, 3 hours?
3 hours,
Oh.
yeah. I guess the point is that you wouldn't use this as an alternative to the police techniques. That's not really what we're aiming at. It's when the police have failed to completely visualize a fingerprint, then that's where the SIMS techniques come in. So we've used it on an anti-terror case, actually, I didn't mention that. We were actually able to provide an enhancement, which is really nice, you know, to actually see it being applied in real life and working. Yeah, but it certainly wouldn't replace the very cheap and very fast techniques that the police already have.
Any other? Paolo?
Yeah, thank you very much. It was fabulous to see all the range of techniques you're developing. And I just have a question on the single cell picking. what's the strategy to go from picking to mass spec? What's the sample prep? What's the resolution?
Yeah, so I
can you detect an organelle?
Yeah, that's a very good question. So, you have multiple options. You can, so the cells are collected into nanospray emitters. So you can spray the cell directly into the mass spec. We got some really nice results there for looking at amino acids, for example, that works well. Or what we've also developed in our lab is you can elute the cell from the tip, and we have a little syringe driver that enables us to do that. We've spent a really long time looking at suitable receptacles to put the cell in, because as you say, the sample prep is absolutely critical. And looking at how to do that transfer as effectively as possible. And then you can do liquid chromatography, which opens up the possibility of looking at proteins, looking at different isomers, doing, I guess, much longer, sort of spacing out your runs and getting much better precision as well than we can with nanospray. So it kind of depends on what the user is interested in doing as to which approach you would take. The nanospray isn't actually automated, so you have to sort of manually load each of your cells onto the mass spectrometer every 2 minutes. And so actually, at the moment, the downstream analysis isn't very automated.
Joe, final question.
I was just going to ask how fresh the samples have to be, because it'd be interesting to see how much information you get from like historical samples and things like that.
Yeah. For which technique?
The fingerprint.
For the fingerprints. Oh, okay. Sure. Yeah. So that's a really good point. We've analysed fingerprints that are several years old, and still been able to see ridges and information. I guess it depends on the purpose of the analysis. But people have been able to develop fingerprints with ninhydrin that are 50 years old. So there's still some information there. I think it will be surface dependent, it will be storage dependent, it will be donor dependent. But it's not impossible, for sure.
Okay, if no further questions, then Melanie's around all day and going to the dinner tonight for the speakers. So ask her questions in the over lunch in the refectory, I guess. So thank you very much. Can we thank Okay, so the next two speakers, I think we have to give a big round of applause because they've stepped in at the last minute due to unavoidable absences of the original speakers. So we're very pleased that we've got the next speaker is George Heath from physics, who's a Bragg University academic fellow, one of the first 3 Bragg fellows. And George is going to tell us something about his developments in our Wolfson lab, actually, with the high-speed AFM. Thanks, George.
Thanks. Is it on here? Sorry. Okay. Thanks, Rick. So it's a pleasure to be speaking at this event. So I will be speaking about high-speed atomic force microscopy, and hopefully by the end, these images will make a bit more sense. So I am based in physics. I'm a physicist, but I look at biological materials. My focus is on proteins, typically membrane proteins, and I like to think of these proteins as kind of nano machines that have lots of different functions, very, very sophisticated, but they're also very, very dynamic and structurally complex. And like any kind of machine, some machines, you can have them so they respond to different triggers. So it could be another molecule binding to a protein, causing a change in shape of that protein. They can respond to things like force. So our sense of touch is down to a specific protein called piezo 1 or piezo 2. And these have very specific molecular architectures that allow them to sense force or respond to lots of different triggers. But that also makes them difficult to study, particularly in kind of structural studies. So if you try and do something like X-ray crystallography, whilst applying one of these triggers and your protein's flicking between different states, it's going to be very difficult. Or techniques like Cryo, you've frozen your sample, so it's no longer dynamic and undergoing its function. So you only get a snapshot. So there's this disconnect between the structure and the dynamics. So a lot of the time people have to kind of infer between two different shapes and how's that gone from this shape to that shape. But wouldn't it be nice if we can actually watch the structures change? And so the technique that we use and we are developing is high-speed atomic force microscopy. So this is a scanning probe technique where we oscillate a tip, scan it across the surface, and basically feel with very high resolution the surface features. And high-speed AFM at the moment can get down to around 50 frames per second. This video here is 5 frames per second of a single protein. And depending on the size of the tip, hopefully you might be able to get down to 1 nanometer lateral resolution or 1 angstrom in the vertical direction. And in my talk, I'm going to talk about a kind of high-speed AFM toolbox that we've been developing that will allow us to branch into different areas. And one of those techniques is high-speed AFM height spectroscopy, where instead of scanning the whole surface, we position the tip on a particular feature that we're interested in and look at how that moves underneath the tip. And that allows us to get higher time resolution down to 10 microseconds at the moment. Another technique we developed is an image analysis method, a super resolution method that allows us to increase our spatial resolution down to sub-nanometer lateral resolution. And the third technique, which I'll briefly mention, is simulation AFM, where if we have a structure that might have been solved by a static method, we can simulate what that would look like under the AFM and start using that for our image interpretation. So traditional AFM is very slow. You scan line by line, and you can almost watch those lines being kind of printed on the screen, kind of like the original internet if you have images coming very slowly. There are some AFMs which go a bit faster, fast scan AFMs, and if you really push these, you can get down to an image every kind of 10 seconds. But it was the development of high-speed AFM that really allows AFM to start looking at molecular dynamics. So this is one of the famous papers from the first developments of high-speed AFM, and this is an actin, a myosin molecule, a molecular motor walking along an actin filament. And these are used throughout our body to move things around and to follow different tracks. They consume ATP as they go, but they can, with this movie, they could actually see how the steps are being made and how the kind of feet are moving around. And in Leeds, in the new Wolfson imaging facility, we now have a high-speed AFM since last year, and we've been starting to look at some interesting biological systems. And the first one was a collaboration with the Astronomy Centre members and some collaborators in Edinburgh. And they were interested in how this protein called BRCA1, which stands for breast cancer associated gene, and people with mutations in this particular protein have high risk of breast cancer. So it's really important that we understand it. And this protein is thought to be, well, it is involved in DNA repair. If there's a double-stranded break in DNA, this protein comes along, binds to the DNA, and helps to catalyze a repair of that breakage. But it's a very floppy and dynamic molecule, so they've been trying to solve the structure and understand how it binds to these DNA, which is wrapped around these histidines here, for some time. But they can't get a good structure because it's in lots of different conformations. It's got these arms that just flop around, so it's very difficult to look at. And the mechanism for how it binds to these nucleosomes is not well known. So we put it under the AFM, This is just the nucleosomes, those two blobs here with DNA wrapped around them. And we can see lots of pairs of nucleosomes under our images. We can boost the resolution with the localization AFM method and start to see some of the DNA that's wrapping around. We can do some simulations to check, is that what we might expect, depending on the size of tip that we suggest. And then if we just look at the Bragg BAD1, this dynamic molecule, we can see lots of molecules bind to the surface, and they're in all sorts of different shapes. And each row here is a single molecule over time, kind of flopping these arms around. And then if we have both of these in solution, so we have the dinucleosome on the surface, then we add the Bragg BAD1 molecule, and get lucky enough to find a moment where the Bragg BAD1, which is going to be here, binds to the nucleosome. We can look at how it interacts with the nucleosome. So this is just overlaid to show our interpretation, but we can see, hopefully, that initially it's kind of bound to this side and the center, and eventually it reaches across to the other nucleosome, detaches, reattaches, and eventually kind of flops off. But for the first time, we could see what the biologists thought was that it would just bind to one of these nucleosomes and kind of clamp around it. But we can see that it can actually bridge across two nucleosomes, and it does it dynamically. And that fit with a lot of their biochemical data as well. And you kind of think of it as Elvis's legs. In the first few frames, you have these kind of legs that are kind of flopping around a bit there. And these are just some stills from there, where hopefully you can see binding to the middle, stretching across, and one arm detaching at the end there. Okay, so that was high-speed AFM. This high-speed AFM height spectroscopy, so normally we'd scan the whole surface. We can push the time resolution just by scanning one line and produce these chimegraphs where you have height in one direction and time in the other. And that allows us to get 1 millisecond while still maintaining some kind of view of the surface in multiple positions. And then the height spectroscopy technique, we have used it to look at a membrane protein called UMP-G, so it's in the outer membrane of bacterial membranes, and it has this flexible loop that pops in and out to kind of block it and open it. And how often it closes that pore depends on the pH, so that the cell can release or allow intake depending on the outside conditions. And we're able to park the tip directly on top of this pore and get out these nice height traces where we can see this kind of two-state behavior where you're flicking between this open and closed. So this is the first time this has been observed structurally. You can observe indirectly if you have this in a setup where you're detecting flow through. So if you have electrical measurements, you can easily detect the current through this pore, but here we can also detect the structural changes. So what about the spatial resolution? So the AFM tip is probably the major factor that limits our resolution. So we scan this big tip across the surface, and we get what's called a tip convoluted image. So everything becomes kind of blurred laterally, but not in the vertical direction. And this is on the scale of kind of 1 or 2 nanometers on the end of this tip. You can get bigger tips, and often we do have bigger tips, although we'd like as sharp as possible. So one idea is that if you have a high-speed AFM movie, you could just average frames and you might improve resolution. If you do that, you just get rid of the background noise, but you still have the problem of this tip convolution. And you can see the challenge that we have if we run some simulations. So this is the surface of a membrane protein called aquaporin Z. That kind of atomic scale, so each of these blobs is an amino acid. And if we run simulations with different tip radii, you can see as the tip gets bigger, the image becomes more and more blurred. And then if we take into account the kind of pixel sampling that we would normally have as we kind of scan across the surface, you can see that it becomes difficult. And then if we add the typical background noise, it just becomes a blurry mess, and that's what you might normally see when you're trying to get high-resolution AFM images of a single protein on the substructure. So if we have a nice sharp tip, this is the kind of image we might expect to get, and we want to kind of recover the information back to the original structure, and that's kind of our challenge. So we took ideas from super-resolution microscopy from the fluorescence field, where this has been developed for quite some time now. You have fluorophores coming on and off, and rather than having the whole surface lit up, if you have them individually lighting up and turning off, you can very nicely locate their original positions because you know what one fluorophore should look like. So it's a localization method. And eventually you reconstruct a high-resolution image instead of the whole blurry thing. With AFM, we don't have any fluorescence. We do have a surface, and this surface with biological systems we know fluctuates. So we have these thermal fluctuations. This is the surface from the side. This is the simulated AFM image, and what we do is we localize those height fluctuations over time, and eventually, in the same way, reconstruct an image based on those fluctuations. And this is the localization AFM method. So we can kind of compare, if we were just to average that video, we just get this kind of blurred feature. The localization, we pick out the 5 points. We can look at the standard deviation, because these are fluctuating features. And you do get some nice signal at the edges and maybe in the middle, but still nowhere near what is actually there. And if we apply this to some real data, so this is real data of that aquaporin Z molecule I showed before. We are localizing the features over time. We also get out the height information for each of those localizations. We can reconstruct this high-resolution image, which nicely matches what has been seen by X-ray crystallography. And we get down to about 4 angstrom for this particular protein. But it will depend on the behavior of the protein itself and how good the images are to begin with. And we've done this for a few different proteins, different membrane proteins. So this is an Xin5 involved in kind of cell membrane repair. This is a transporter. We've looked at 2 different pHs, so we could get the structures or the surface structures at these 2 different pHs and see how there's a conformational change, which is thought to be involved in how this channel actually functions. And because we are limited to the surface, we do have to kind of think about what these changes translate to in the rest of the protein. If one feature on the top moves, how's that going to translate down the alpha helices? And the final example here is of this OMP-G protein I showed earlier with the height spectroscopy, where we were detecting the loops moving in and out. We could actually visualize that if we take lots of images of the open and lots of images of the closed, and it matches nicely where we expect the loop to come in. Okay, so in the last part of my talk, I'll going to briefly mention some analysis method we've been developing, or other tools. So one of the first things we realized that when we got this new high-speed AFM is we're collecting all this data at 10 frames per second or up to 50 frames per second. How are we going to analyze it all efficiently? Because the manufacturer's software isn't necessarily built for high throughput, it's just built to get the data out. And we're getting millions of pixels per minute. And as you saw with the localization AFM methods, we care about a lot of those pixels, and we don't want to not pay attention to them. So we've developed some open source software called Nanolox with the idea that we can take any raw AFM data from any AFM, particularly for us, there was no tools for high-speed AFM, and apply lots of the standard kind of leveling and pre-processing, and that we would also integrate this simulation AFM so that we can compare your real data to simulations of proteins and things, and get out things such as area analysis, particle detection, the localization AFM, which I mentioned, and get out some quantitative information. And I don't know if I need the mouse for this. Is there a mouse? Or maybe it just plays? No, it doesn't play. And this is just a brief demo of the user interface where we're loading in different file types, automatically leveling the images, playing the videos very quickly, and applying lots of different features. I'll speed this up because I'm running out of time. No, maybe I can't. Okay. It's a couple of minutes long. We don't have a couple of minutes. But we have lots of different workflows that we can combine and allow any user to perform AFM analysis much quicker, and we'll still be adding lots of features to it. And so one of those analysis workflows was the localization AFM. So these are DNA origami. So we pick out all the origami, recognize where their positions are, do lots of alignment. We can do this within less than 5 minutes. Do single particle analysis, so localize the fluctuations, and get out super resolution images at the end, which are much higher resolution than the individual frames. So I'll finish there just to show the comparison between the two. And say that the height spectroscopy allows us to get 10 microsecond resolution in time. The localization AFM allows us to get 4 angstrom spatial resolution, and this Nanolox software should allow us to automate AFM analysis and high-speed AFM analysis. And thank you for listening.
Thank you, George. That's amazing. Megan.
Do you think you could get the acoustic mode spectrum out of an individual protein?
I guess theoretically, we did try at one point to position this on an ATP synthase, so it has like a rotary motion, so that we could get out the fluctuations and kind of almost read it like a record player was the idea, as a nice idea, not very scientific, because it's already known how kind of fast it rotates, but it is, is that similar? Is that answering the question? Yeah.
Any further questions? Lorna?
That was really incredible, George. Thank you. You very briefly mentioned that the dynamic information that you get on the surface of the protein, I think you said you could then start to think about what would happen in the rest of the protein. Could you say a little bit more about that, how you translate that information?
Yeah, so we haven't done it yet, but for that particular protein, it was about, maybe we have kind of slightly done it. You can use that information in molecular dynamics simulations, so you can kind of steer your molecular dynamics simulations with these different structures, so they could fix the protein in one position that you've seen in the AFM and see if it will naturally go to the other state, and use those kind of surface features as constraints on the dynamic simulations. Or you can kind of hand wavy, say this has moved over here, this might have moved here. If you have images of both sides, then you can kind of try and translate what's going on.
Question at the back. Probably have to be the final question, I'm afraid.
Hello, my name is Victor. I have one question. When you presented the videos of the molecules, there, in the time was passing, there were some dynamics on it. Is it because of the dynamics of the molecules, or is the register of the technique to analyze these molecules? Because according to the, let's say, the energy of the electron, I will expect a higher frequency in those dynamics, but it's probably the recording of the molecules. But I'm speaking about the changes, the slight changes in the molecules.
So is this the red trace, the height spectroscopy?
Yeah, just in the videos you show, the molecules are not static, are fluctuating a little bit, are changing. Is it because of their recording method, or is a real state of the molecules?
So it's a little bit, we think it's a bit of a combination. So obviously we are kind of tapping on these molecules, but naturally in molecular dynamic simulations, they do fluctuate in similar ways that we see. In general, we do see if there is a specific conformational change that happens. When we scan by AFM, the kinetics are a little bit faster than another technique such as FRET. And we think this is because we're inputting a bit of energy. It's almost like you're effectively heating it up a little bit. But you do try and maintain a very low force, and it's kind of on the single kBT or a couple of kBT per tap. So it's supposed to be hand wavy, similar to the surroundings, but yeah. The question of are we influencing the sample is a very common one, and we most likely are a little bit, but we try and minimize it.
Just giving it a pat on the head, aren't you? Okay, well, thanks very much, George, for that update. It's really exciting to see what's going on in the new Wilson lab. Again, a big thanks to our final speaker in this session, Stephanie Foster, who's a postdoctoral researcher in, she's been both in the School of Chemistry and the School of Chemical and Process Engineering. So Steph's going to tell us something about carbonation of calcium hydroxide using cryo and liquid cell transmission electron microscopy.
Hi everyone, I'm Steph, and there we go. I'm going to give you a very quick run through of my project on, as Rick said, the carbonation of calcium hydroxide using different TEM techniques. So what is the carbonation reaction? It's quite a straightforward reaction where we have here this slurry of calcium hydroxide, and we are bubbling through our CO2, and we're ending up with calcium carbonate, and in particular we're ending up with calcite. And it's this scalenahedral form that we are ending up with, not nice little cubes that you might expect for calcium carbonate. And this is an important reaction because this is the industrial way that they mass produce calcium carbonate, which is used in a multitude of different things. So the point of our project then was to look at the reaction mechanism underlying that. So is it a solid state transformation, or is it a dissolution reprecipitation pathway? And in discovering that, we can also see how important is the state of our starting material, that calcium hydroxide. So we decided to use TEM to try and tackle this, and it's really good for looking at the early stages of crystallization. We start off with our nanoplates of calcium hydroxide. So these are our really thin plates that are about 20 nanometers in thickness, so it's ideal for looking at using TEM. It's not too thick. And we make a suspension of this in ethanol that can then be put onto a grid, and we'll do the reaction on the grid. So we can just allow CO2 in the air to carbonate our calcium hydroxide. And we will just plot that at different time points, and we'll do dry TM on that, so quite a straightforward experiment. But obviously that's not an ideal way of pausing our reaction at different time points, because the CO2 in the air, and after you've plotted it, there's nothing to stop it from necessarily carbonating further. So this is where cryo TM comes in. And if we do full cryo, we can also get an added bonus of being able, for our sample, being able to tolerate a higher beam dose for longer, so we can look at it more before there's any kind of beam damage occurring. And finally, what I'm going to talk to you about is using liquid cell to study the reaction. So looking at it in the solution state and really identifying the dynamics of the reaction, which we don't get with our static techniques. So to start off with, we did some dry TEM, and you can see our earliest stages of reaction. Remember, this is not being kept in a specific CO2-free way. We can see that there is some dissolution occurring already on our basal faces. So you can see here that these gaps in our central face of the hexagonal plate are appearing. And we can see after even further reaction here, we end up with lots and lots of these kind of like holy hexagonal rings appearing. And some of these are starting to look a little bit more globular. They look a bit darker with their densities. And we know from doing electron diffraction of these that these are amorphous structures. And indeed, we do our elemental EDX mapping here, and we can pull out all the elements there, showing us that we've got ACC, this amorphous intermediate phase. So the fact that it's retained its hexagonal shape tells us it's a pseudomorphic replacement reaction, so the morphology is being retained throughout. And we can look in our textbook and we can identify this kind of reaction process, a coupled dissolution precipitation reaction. So we have this dissolving plate that we end up with this supersaturated boundary layer, and we'll end up with ACC on the plate and around the plate. And then this is very reminiscent of this dissolution precipitation reaction pathway. So doing our cryo TEM, we actually did some tomography, so because we can tolerate that high beam dose, we took lots of pictures over a set of angles, and we can put together these tilt series. So what we can see here is just the very clear hexagonal morphology. We've got really nice 120-degree angles between our edges, and also the amorphous material that we see kind of clustered around it. Because we're tilting around the sample, we can see that that is connected. It's not just above or below our sample. Looking at it a bit more with dry TEM, we actually added some water to our grid. So we know from the literature that that catalyzes the reaction even faster. So we wanted to see the later stages that weren't just full of calcite at the end. So we know that by adding a bit of water, after 2 minutes, we see something similar to before, this kind of mass of amorphous, holy hexagonal rings. But we also see these crystals. And this is our scalenihedral calcite at quite an early growth stage. We can see that this is, when I say scalenihedral, it's this kind of structure that's fatter in the middle and thinner at each end. And it's at these ends that we see these crystal needle-like protrusions emerging from. So growth is occurring along that C direction. When we do some electron diffraction of this, if we look at our yellow area here, so if my aperture is over here, we can see that we get these, we're focusing just on these needle-like crystallites. And then this is the matching diffraction pattern. It might be a little bit hard to see, but we have these diffuse arcs in our diffraction, just what we would expect from having multiple nanocrystallites. But when we move into the central area, so this green kind of ring that you should be able to see, we still obviously have some of our crystallites that are the needle-like crystals in view. So we see that in our pattern. But what's interesting is that we also see this really strongly this overlaid really strong single crystal pattern. So that shows us that this crystal here is a single crystal. So after seeing that, we were thinking, what does this tell us about our growth then? It indicates to us that perhaps we're starting with these kind of aligned needles, and then as it's growing, we're undergoing a consolidation process, some recrystallization, so we'll ultimately end up with a single crystal. So back to our cryo TEM tomography. When we did this, it was really nice because we really could clearly see this structure here and independent kind of crystals here that are all around about 20 nanometers thick. And these are all aligned in this common direction. So it's not this larger crystal that we see before, saw in the dry TEM. It's actually consisting of individual needle crystallites. So moving on to liquid cell. So I'm not going to run too much through the actual setup of that, but feel free to ask me later if you have any questions. This is the holder itself, and it's a clamped kind of design. So we have 2 silicon nitride chips with windows in them. We will load, as we did for the TEM grids, we will load a droplet on one of these plates. We will assemble it as normal, put it in the TEM, and then we will, I should say that when we assemble it, we put ethanol in it, so it's kept from any reaction. Then we put it in the TEM, and then we displace our ethanol with our water. And then we can watch the reaction unfolding. And we get all these nice videos. So hopefully you can see nicely, we've got 2 hexagonal plates here. And then as our reaction unfolds, we can see dissolution occurring on the basal faces again. And what's really nice is that we can see, there we go again, or not. Okay, maybe not But what we could see was we have these really nice outer edges that are preserved, and even in our liquid cell we could see that. And we could also see some of our ACC kind of around that being formed. So looking at it on a different day, in a different set of experiments, we could see that we have more dissolution occurring. And then at the end here, what we had was a bubble come into our field of view. So that's one of the challenges of liquid cell, that sometimes every now and then a bubble will pop in. But what this does show us, with this nice little moment of HD resolution, because we haven't got the liquid there to deflect our beam more strongly, we get a nice HD moment where we can really see how amorphous our partially dissolved plates are. And then finally, we actually attempted to rehydrate the cell here. And although we can see lots of our ACC dissolving, we weren't really thinking this would work too nicely, but after we'd walked away from the microscope and we were going through our data, we could see in this corner that as things around it were dissolving, what we could see more clearly pointed out here is actually the emergence of these needle-like crystals, which is what we saw previously in our dry TM and our cryo TM. So we have actually had some nucleation of our calcite. So I don't know how we're doing for time. I'll give a quick run through of this mechanism then. So using all these different TM correlative methods, we've been able to extract all these details on this mechanism. So I think hopefully by now I've convinced you that it's a dissolution reprecipitation pathway. We have our dissolution, of the faces, mainly on this basal face. That's giving rise to this pseudomorphic replacement with ACC, and we end up with this pseudomorph where the hexagonal state is retained. So obviously it is important, the state of our calcium hydroxide at the beginning, because it's a pseudomorphic reaction. And then we've also been able to look more at the growth. So the main interesting thing for us is seeing that this is not quite a basic looking crystal to us, that it has got all these kind of nano features, these aligned crystallites of these needle-like kind of shapes, and that this must undergo some kind of consolidation process to end up with what we see in like our SEM images, if you remember from that earlier slide. So that's all from me. Thanks to everyone that's been involved in this project and to our funders.
Thank you, Steph. Time for a couple of quick questions.
Everyone's hungry.
Neil.
Thanks, Stephanie. Very nice imaging there. Can you say anything about what happens in this process from the amorphous to the crystalline? Is there some secondary nucleation process, and is that registrated between the original calcium hydroxide crystals and the calcite?
So you mean, is there between the precipitation of ACC, or specifically from the hydroxide to calcite?
Well, from the ACC to the calcite. Yeah. So is it, do you have a working model whether it's complete dissolution and recrystallization, or are those nucleation sites connected to where the original calcium hydroxide came from?
Yeah, so from our data, we're seeing that it's dissolving and then reprecipitating. We're not seeing like any kind of nucleation occurring on an existing patch of ACC. I think that kind of fits with the literature as well of what we know about ACC. But obviously TM, we are looking at certain parts of it, so we don't know. There might be some areas that we do get a bit of secondary nucleation, like you say, or nucleation from the ACC, but we haven't captured areas that tell us that.
Okay, thanks.
One quick one from Sandra.
Thanks a lot. So we see, thank you. We see when we look at rocks and we try to figure out if millions of years ago we had dissolution precipitation reactions or actually diffusion changing the chemistry. One of the things we've found out now is that we get slight misorientations within the rim of replacement, which we think are replacement. So that could kind of would be an effect of your little strands that actually then kind of consolidate. So that would be, is that right? Do you know, is there a slight misorientation in the orientation of these strings you have? Can you see that?
What, sorry?
The arcs.
So they are slightly
Yeah, they're not completely perfectly oriented together, like you say, because of the diffuse nature of that. We haven't specifically looked at it that much because we've been quite focused on the dissolution part of it. So with that, we've been able to do some high-res TM and lattice imaging of that. So that is probably our next point, actually, is to go back into the... it has to be cryo, really, for that as well, because I think we're actually, with the dry TM, causing recrystallization. So it looks more like a single crystal with that, but that's definitely something that we've picked up on for the cryo, to go back into that data and try and look exactly at those needles and be able to answer that a bit more convincingly, hopefully.
Yeah, that'd be really interesting. I think you would see it, yeah. Thanks.
Great. Well, if no further questions, then we're running over time slightly, but meeting into lunch and posters. But Andy, do you need to say anything?
No, I think that was a fantastic first session. Thank you for all the speakers, and thanks, Steph, again. As Rick indicates then, it's time we're going to move over to the refectory where lunch is being served. We've got 65 posters to see and a facilities exhibition as well. So please do head over that way. My lovely colleagues will direct you if you do not know where. Thank you. Brilliant. Lovely talk.
Thank you.
Yeah. So we're just running a little bit behind schedule after the morning session, so we're just going to carry that on a little bit. So talk amongst yourselves while everyone else comes back. It'll be about 5 or 10 minutes.
Right, so good afternoon, everyone. So welcome back to the afternoon first session. So my name is Liu Chuanyang. I work in School of Mechanical Engineering, and I also manage one of the Royce and Bragg facilities at the School of Mechanical Engineering. So session 2 is about functional surfaces. So we have 3 speakers. Our first speaker is Dr. Gareth Hinds from the National Physical Laboratory. So I've been given some introduction from Gareth that he's a senior NPL fellow and a science area leader in the electrochemistry group. And he's leading a team of scientists undertaking groundbreaking research in corrosion, electrochemical energy conversion and storage, and analytical electrochemistry. Gareth is a fellow of the Royal Academy of Engineering and holds visiting professorships at various universities. And he's going to give us a talk about towards more representative corrosion testing of fuel cell and electrolyzer bipolar plate materials. So let's welcome Gareth.
Thank you, Liu. Good afternoon, everyone. It's a pleasure to be here at the Bragg Exchange. There is one link, at least one link, with NPL of William Henry Bragg, and that is that his son, Lawrence Bragg, was briefly director of NPL from 1938 to 1939. And to this day, we still have a road in the site called Bragg Avenue. So we do have that link. I'm here today to talk about corrosion, and specifically corrosion in electrochemical energy conversion devices. So put your hand up if you know just a little bit about a fuel cell or an electrolyzer and how they work. Okay, actually a fair few, and I'm finding this more and more as time goes by. It used to be that only one person would put their hand up, or maybe even no one. So it's good that the general awareness of fuel cells and electrolyzers is increasing. So I am aware that this is a very broad conference covering materials in a whole host of applications. So I'm going to just give a general introduction for those of you that may not have heard of fuel cells and electrolyzers and how they work. So first of all, how a polymer electrolyte membrane or proton exchange membrane fuel cell works. It's a 2-electrode device, an anode and a cathode, separated by a proton-conducting membrane. You supply hydrogen to the anode, you supply oxygen to the cathode, and you get 2 separate reactions that, when put together, convert hydrogen and oxygen into water with electricity as the byproduct. So hopefully this pointer will work. Yes, great, it does. So we've got hydrogen gas supplied to the anode catalyst. This catalyst is platinum decorated on high surface area carbon. We have the same catalyst typically at the cathode where we supply the oxygen. And this material in the middle, the membrane, is the key material. It conducts protons, but it's insulating to electrons. And that means that when we break the hydrogen into 2 hydrogen atoms and electrons, the electrons are forced around the external circuit. The protons migrate through the membrane, and then they react with the electrons and the oxygen from the air at the cathode to form water. But crucially, the electrons give us power. So this is a very simple electrochemical device. You take hydrogen and oxygen, and you generate an electrical current. So this type of fuel cell is what will typically be used in a car, or a truck, or a ship, or an aircraft, when we use hydrogen as a fuel. And in the decarbonization of the energy system, hydrogen is an important component because there are parts of the energy system that are very difficult to decarbonize, particularly industry processes, particularly heavy-duty transport, where batteries become prohibitively heavy. So this is a device that you're going to see more and more of in the future, particularly for heavy goods vehicles, for trains, for ships, for aircraft, and the other part of this is where we produce the hydrogen from. At the moment, most of our hydrogen, 99-point-something% of it, is produced from fossil fuels. So while we can sequester some of that CO2 in what's called blue hydrogen, that's not a long-term sustainable process. So green hydrogen is where we use electrolysis of water to produce the hydrogen. Then when that hydrogen is used in a fuel cell, yes, there are quite a lot of efficiency losses, so we'll lose more than half of the energy. But when we have wind curtailment, we may as well use that wind, generate lots of hydrogen, store it for long periods of time, and then use it either by burning it in a combustion engine or by using it in a fuel cell. So I'm going to talk about the electrolyzer device, which is pretty much this device working in reverse. So again, it's two electrodes, an anode and a cathode. Everything's the other way around now. We're taking water and we're applying an electrical current to it to produce hydrogen and oxygen at each electrode. This is something you may have done in a chemistry class in school, taking water and splitting it into its constituent gases. So we generate oxygen on the anode. Again, we have this same membrane material that conducts protons but not electrons. This time we're supplying our electrons from an external power supply. We're splitting the water into oxygen and the protons are then migrating through the membrane, combining with the electrons to form hydrogen gas. And that hydrogen can then be stored. One of the big advantages of hydrogen is you can store it for long periods of time. Anyone who's charged their phone, I was about to take it out of my pocket, but I don't have it on me. Anyone who's charged their phone and then left it for a few months will know batteries are not a good way of storing power for long periods of time. They're great for putting electrons in and out, in and out. It's like a muscle. They can do that for long, many, many cycles. Seems to be skipping ahead. But hydrogen, you can store it in a tank, you can store it in an underground cavern, and it will still be there when you come back, okay? So for heavy-duty transport, for long periods of time, seasonal storage, hydrogen fits that niche. I would never suggest using it for small vehicles, for example, or when you need to charge and recharge every day because of the efficiency losses. But that's the trade-off. Batteries are much better for small cars. Hydrogen are better for long periods of time and much heavier modes of transport. So having introduced you to those two devices, I'm now going to look at the cost, because really that's the key thing. They're still too expensive. So this is a breakdown of the cost of a fuel cell stack, which is what we call a stack of lots of those cells in series. So there could be, in a vehicle, there could be hundreds or even thousands of cells stacked up to produce enough power. And the same for electrolyzers. If you want to generate a small amount of hydrogen, you just need a few cells. If you want to produce enough hydrogen for a refueling station, you're going to need hundreds or thousands of cells with a massive area. And there's a lot of engineering that goes around that. You need plates, you need membranes, you need gaskets, you need balance of plant, and this can become quite complicated. And all this adds to the cost. But the topic of my talk today isn't the electrodes, it isn't the balance of plant, it isn't the hydrogen, it isn't any of the conditioning. It's actually this particular component. So it exists in both. It's called a bipolar plate. And you can see it makes up a significant fraction of the cost of both technologies. And the main function of the bipolar plate is just to conduct electrons within the stack. So they're typically metallic. They also provide mechanical stability of the stack, and they make up the bulk of the volume and the mass of the stack. So typically, this is what a bipolar plate looks like, these gray units that repeat throughout the stack. And the reason they're called bipolar is that you have these membrane electrode assemblies between each bipolar plate. So each bipolar plate has an anode side and a cathode side. So hence it's supplying, if it's in a fuel cell, it's supplying hydrogen to one side of one cell and oxygen to on its other side to the other cell. And there are a whole load of challenging material requirements. So you guys out there are working on a whole range of different materials. We're probably going to need expertise of every one of you to solve all of the problems that these materials have for these components. So I'm not going to read out what all the things a bipolar plate has to do. You can read them for yourself, and you can probably relate some of your expertise to what might be needed to improve all of those parameters. But I'm interested in the corrosion resistance of the bipolar plate. So you've seen that there's water there. These things operate typically at 60 to 80 degrees Celsius, and the membrane that I mentioned is acidic. So temperature, acid, and water. Put a metal in that, it's not a good thing, okay? So we have to be really careful about corrosion. And in the space program, when this technology was developed, actually graphite was used for bipolar plates. And graphite is the perfect material because it's very highly conductive, and it doesn't form a very insulating surface oxide like a metal like a stainless steel does. However, graphite's not a very strong material, so you have to make it quite thick. That makes it very bulky, too big, and sometimes too heavy. When you use a metal, you can make it very thin, and that makes these stacks smaller and lighter. The problem with a metal is it's going to corrode, and its contact resistance is going to increase with time as the small oxide layer on the surface builds up, even for a stainless steel. Now let's look in more detail at the operating conditions. So typically, here's our bipolar plate, one on each side of the cell. We've got a porous transport layer or gas diffusion layer, which typically consists of a carbon paper. And then we've got our electrode on each side. So here's our platinum nanoparticles decorated on the gray high surface area carbon. And then in between these two electrodes, we have this membrane, this acidic solid membrane material that conducts protons, but is insulating to electrons. And typically, these are the voltages that we see during startup and shutdown. So the local potential at the bipolar plate, it's assumed in the academic literature and by the manufacturers of these devices that the local potential of the bipolar plate is equal to the potential at the electrode. So during startup and shutdown, you might see 0.7 volts on the platinum and up to 1.5 volts on the cathode electrode. And the common assumption before we started doing this work at NPL was that we would see the same potentials on the bipolar plate, which, for a metal, 1.5 volts is pretty severe in an acidic aqueous environment. The same with the electrolyzer, but it's even worse for the electrolyzer, because here you're applying an additional voltage to the stack. And typically, at the cathode, because you're generating hydrogen here, it pretty much behaves like a hydrogen electrode. So it will sit at 0 volts on the thermodynamic scale. The more challenging electrode is the anode. And here, you need 1.23 volts to split water, right? You need a little bit extra just to get over the activation barriers and some of the losses. So typically, in an operating electrolyzer, you'll be sitting at around 2 volts during operation. And the assumption was that you would also see 2 volts at the bipolar plate. And that means you need platinum-coated titanium. Platinum-coated titanium is a horrendously expensive material. It's difficult to machine, and both of those materials, the substrate and the coating, are expensive. But this is what people are using. In pretty much every anode bipolar plate material is platinum-coated titanium. And not only that, but because it's a bipolar plate, even though you don't necessarily need it on this side, it has to be all the same plate. So you've got to make everything out of platinum-coated titanium. Really expensive. We set out using our knowledge of corrosion. So my group has a long background in corrosion, and only in the last 20 years have we started looking at fuel cells, electrolyzers, and lithium-ion batteries. But we knew straight away that this assumption was just not correct. So we set out to validate that hypothesis, and we used this quite neat arrangement called an in situ reference electrode. And what we're doing with this reference electrode, it's a regular, commercially available hydrogen reference electrode that you can buy off the shelf. But the neat part was we used this tubing made out of the same membrane material as inside the fuel cell or electrolyzer. So this is a proton-conducting solid And this tubing is only available from one supplier in the world. They use it for water purification technology. It's available from this company in New Jersey. And it works really well as a solid salt bridge for this application. And it's an ideal salt bridge. It doesn't contaminate the system of interest. It provides a perfect ionic conducting path between the reference electrode and the point of interest, wherever it might be inside your cell. So effectively, we're then able to measure the local potential at the bipolar plate in an operating fuel cell or electrolyzer. Now, we did the fuel cell work first in, I think, about 2013 or 2014, and only recently have we applied it to electrolyzers. If we'd done it the other way around, then I might be looking at being quite a wealthy man. But unfortunately, I did it the other way around and I published it. So I'm not bitter, don't worry. But as you'll see, the implications for fuel cells are more around how we do the testing and how we qualify different coatings and materials for the bipolar plates. The implications for electrolyzers, on the other hand, are we can get rid of a lot of expensive materials and put in much cheaper ones. And that could halve the cost of green hydrogen technology. So that's a very commercially important discovery. Unfortunately, I couldn't patent it because I'd already published it for fuel cells. Anyway, I'm just happy that we're able to contribute. So let's have a look at the original measurement for fuel cells. We took an uncoated stainless steel bipolar plate. You can see this is how the gas is distributed to the fuel cell through these serpentine parallel channels. We drilled a hole for the reference electrode tube, just one hole. We just want to measure the local potential. So we're measuring the corrosion potential, which is what we call the local electrode potential of the stainless steel during the operation of a fuel cell. And we want to show that it's not going to be the same as a higher potential that the electrode is experiencing. And indeed, it worked. This is the cathode potential. You can see we varied the cell potential and therefore the potential at the cathode at lots of different levels, but the steel potential just effectively stayed at open circuit. So it's completely decoupled from the cell voltage. And this was, you know, it took a lot of convincing of people that this was real, but it turns out it is. And actually, I'm going to a PhD defense in Stockholm at KTH on Friday, where a PhD student has, amongst other things, spent the last 4 years proving that this hypothesis is correct and does work at stack level. So that's going to be really nice. But had a lot of arguments with reviewers and with people at conferences about all of these concepts. When you put a microporous layer in, I'm not going to dwell too much on this, it's a bit of a detail, but even during startup and shutdown, when you get these huge spikes in potential at the electrode, you don't see anything at the bipolar plate. So the bipolar plate effectively doesn't know that the electrode is there. So that really high potential that could be very corrosive is not transferred across to the bipolar plate. The same thing for an electrolyzer. Okay, this time we take an electrolyzer cell, which, as you recall, is the same device working in reverse. Here we're supplying water to the cell and using electricity to split it into hydrogen and oxygen. And we've used the same reference electrode approach with this salt bridge terminating at the component of interest, which in this case is the current collector. We call it a current collector when it's just one single cell because it's not a bipolar plate, but it's effectively simulating the surface of the bipolar plate. And again, we showed this decoupling. So this would be roughly the potential that you would expect the anode to sit at, the anode electrode, close to the cell potential. And the anode current collector is sitting at open circuit. And there's a huge drop in potential from about 1.6 volts down to about 1 volt, or even below. And this is proof that just like in a fuel cell, the anode current collector is independent of the potential at the anode. So why is that? The reason is, and this comes from the principles of cathodic protection that are used in protecting steel structures in the ocean, for example. In order to transfer a local potential from one metal at one location to another, you need two things. You need an electronically conducting path, which they usually have, and they do in these devices. It's perfectly well conducting. The other thing you need is an ionically conducting path. And in seawater, that's made up of the salt, that's dissolved in the sea, and that can give you a reasonably good, what we call throwing power, to transmit the voltage, say, from a sacrificial anode onto a steel structure. It can go centimeters or even meters. However, the environment here is ultra-pure water, so it has very little ionic conductivity, and that means that any potential that you see at the electrode is only transferred a few hundred microns in that environment, at best. And that's the central concept behind this, that when you have a very pure electrolyte, your potential doesn't transfer very far. So effectively, the bipolar plate, which is sitting several millimeters away from the electrode, is unaware that there is a high potential in the local neighborhood because it can't be transmitted through the pure water because of its very poor conductivity. So I think in the interest of time, I'm going to skip over the more recent work that we've done on the porous transport layer, but I think you can see here some of the modeling that we've done to show that depending on the conductivity of the water in an electrolyzer, this is type 1 water, which is highly pure. When it's slightly less pure, with type 2 water, you can see that the potential persists a little bit further, but again, we're talking just a few hundred microns here. And then when we bump up the conductivity to really high levels with pH 1 sulfuric acid, which you would never see in an electrolyzer, but it's just here to demonstrate the concept, then you see that the potential does persist all the way to the bipolar plate. So if you can control the conductivity to either type 1 or type 2 water, which every electrolyzer manufacturer does anyway, it needs that to work for lifetime, then actually the high potential only exists in this small region very close to the catalyst layer. So certainly for the bipolar plate, you could put any material there. And we would recommend carbon-coated stainless steel. It's already manufactured in bulk, it's cheap, and as I mentioned, carbon is a great surface for contact resistance. We validated the model. I won't talk through it now, but we did some nice experiments that showed that this potential profile is real. But this basically summarizes the work for electrolyzers, showing that we have this high anode potential at the catalyst layer, and then because of this low conductivity of the water in this porous transport layer, we just have a very thin corrosive zone, a few hundred microns typically, depending on the geometry and the electrochemical properties of the different materials. So the bottom line is, this took a lot of convincing, but it's a very simple concept that came from effectively corrosion in the oil and gas industry and was transferred into a new area where the concept was very alien. It took a lot of arguing, it took a lot of rejections from journals to get this work published, but now it's pretty much established as a scientific fact, and hopefully it will make a big difference, not just to the way that we test materials, so particularly for fuel cells. We don't need to test them at such severe voltages when we carry out ex situ screening on materials, but I think even more significantly, we can substitute cheaper materials in a lot of these devices and reduce the cost of the technology, particularly when that is one of the bottlenecks in widely adopting electrolyzer technology for green hydrogen. So with that, I'll thank you for your attention and happy to answer any questions.
Thank you very much. So it's a very, very nice talk. Any questions from the audience?
Get your steps in. You mentioned it was originally platinum-coated titanium.
Yes.
Why was that? I can understand the platinum coating, but why titanium?
So the titanium is there mostly for its corrosion resistance at 2 volts.
Right.
The
platinum's the catalyst on it.
It's not there for a catalyst, it's there for contact resistance.
Right.
So the titanium, if you don't have the platinum there, the titanium will build up a thick layer of titanium oxide that will result in too much loss when a current flows through it. I should say that you say it used to be. Every manufacturer is still using these materials. It will take, I don't know, 5 to 10 years for this new material idea to work its way through the prototype development cycle. So they're stuck with the expensive materials for the time being.
So,
so I just was going to ask about sort of NPL's role, because obviously you've done a really, I love the study you've done there, and I love the comparison with the oil and gas. But I suppose we've got lots of people developing fuel cells, developing this sort of technology. How is NPL's role in actually sort of validating some of the measurements? Because that's what you traditionally do. How's that working in this particular area, not knowing very well?
Yeah, so I think, so first of all, the first thing we've done is passed on this information to electrolyzer manufacturers and, crucially, to stainless steel suppliers. So in one particular instance, we've hooked up a leading UK manufacturer with the leading European manufacturer of carbon-coated stainless steel. And we know that, I can't say any more than we know they're developing the concept. We're not involved in that really closely. What we are doing, though, is we're making the model available. So they quite commonly come to us and say, would this design work? And then we'll model it for them and show, you know, what kind of drop-off they'll get in potential and whether a particular geometry will work. And then, of course, we do testing too. So when they get to a point where they want an independent validation of the stack performance or a single cell, we'll do the testing and the modeling. And when they, particularly for a small company, when they go to investors in the next round looking for funding, it's much more convincing to have an NPL set of data than their own. And that's quite often where we come in.
Follow up then, and for the academic community, is the same sort of thing happen, or is it more the commercial community? Because I can see validating data to go into publications, whatever, and again, the NPL stamp on that is obviously good for a lot of these sort of parameters.
I think when it comes to the lower TRL work, I think that's one of the nice things about working at NPL. We span all of the TRLs, so we will have academics coming to us, usually more validating an idea or a concept rather than an actual device. But we do a lot of that. We tend to work in two ways, though. We'll either, I think if they think it's going to be very commercial and they're quite confident in it, they'll just pay us to do a bit of work to validate it to the next level. If it's a little bit more blue sky and exploratory, we might, and it's aligned with our own interests, we might collaborate with them, not charge them anything, and publish a paper. But we have that flexibility to work in different ways.
More questions from the audience? Right, so I guess we will thank Gareth again for that very nice talk. So our next speaker is Dr. Frederick Pessu from Institute of Functional Services at School of Mechanical Engineering. So Frederick is a lecturer in corrosion, and he has a broad interest in application of a corrosion science in understanding the behavior of a material interface across various of energy systems. So he's going to give us a talk in understanding corrosion interactions in systems designed to deliver clean energy and net zero targets. So let's welcome Frederick.
All right, thank you very much. Good afternoon, everyone. My name is, well, they've called my name already, so no need for that. So I'll be speaking on systems that has been designed to deliver clean energy and meet net zero targets. There's a broad range, obviously. You have different areas of which I'm involved in them, geothermal and carbon capture and all of that. But I'll be speaking mainly on molten salt energy systems. And just a bit of introduction. I like to use this map here, which kind of shows the intensity of direct solar radiation across the globe. And you can see where the solar resources are pretty much in. You have the US, you have in Chile, you have the north of the Sahara, south of Europe, and then also you have in Southern Africa. And then this is where we are. green part where you don't have a lot of sun. But that doesn't mean we don't provide some value in terms of research and development. So, and just to give a bit of numbers, to give context, so you have the potential energy you can get, from solar annually is about 4,000 exajoules. And in 2014, the annual supply is about 573 exajoules. So you can see the huge gap there, and also shows the untapped resources we have. And that just to show how much the world is going to be depending on solar energy in the future. But the key point of this introduction here is just to really say how we can transfer this energy into electricity that we use every day. And one of the major ways to do that at an industrial scale is the concentrated solar power plants. Now, the interesting thing about that is you have a lot of heliostats that concentrate the heat on the tower receiver, and then you require a heat transfer medium that goes to collect all the heat, usually at a temperature of about 1,000 degrees C, and then this is then channeled through to the power block that converts water to steam to generate electricity. So that's where we get a useful solar thermal electricity from. And then off-peak time, excess of these energy is stored in the storage tank, and that is the tap is turned on at nighttime when you don't have a lot of sun. And then it helps to deal with the intermittence of solar energy that you get at nighttime. And then it goes back into the cold storage tank, then the cycle continues. So that is the pathway of the heat transfer medium. And the most common one, you have various types of heat transfer medium to use. You have from oils, and then you have different salt chemistries. And this is where the challenge comes in. These salt chemistries, if you see here, at this point, they are usually low to moderate melting point, and then a high boiling point. That gives you a wide temperature scale for which to use to harness the heat from the sun. And so depending on the chemistries, then you can see you have different boiling point and different melting point, and that tells you the limit of your concentrated solar power plant. So the conventional ones are the solar salts, which are, to come closer. So you have solar salts here, which is the most common one to use. These are basically nitrate salts. The boiling point is about 565, so the plant, the CSP, doesn't work above 500 degrees C, because above that temperature, the salt degrades, and then it becomes very corrosive, and then it starts to heat up, corrode the materials. And because of the huge investment in solar power plants, they are required to last a long time. But when you have the salt being aggressive, being ionic, then you have a lot of challenges. And also the high temperature also is a problem, because without the molten salt itself, you're always going to have a high temperature induced degradation. So that's on the solar side. But more interestingly is the fact that the molten salt is being used as well as a way of conducting this heat to be used for most industrial processes. So you can have it, you can use it for blue hydrogen generation. You can also have it for gasification, biomass processing. You can have it for different industrial applications. You can use it for waste heat recovery as well. So there are some systems where you have a lot of high heat being wasted, then you can use a molten salt to conduct that. So that's on the solar side. So the challenge, I know I mentioned earlier on that we don't have a lot of sun in the UK, but it's interesting to note that most of the investment in Northern Africa, they're from European countries, and in France, Italy, and the whole idea is to use underwater sea cable to take these, transmit your solar electricity to Europe. So there's a lot of investment into billions of dollars by 2040. So that's kind of on the solar side. But most interestingly is that the same process is taking place in molten salt reactors. So you have the MSRs. So you have your reactor core, where you have your fissile material mixed with molten salt, and then it goes through heat extraction circuits. So you're transferring the heat with the heat from the core of the reactor with the molten salt to another intermediate zone with heat exchangers, and then you have that being transferred to the power block. So that's your power generation circuit. So it's the same process of being able to transfer thermal electricity from the core of your reactor to the power block. So the connecting system here is a molten salt for both applications, used as heat transfer and also coolant. And it has to interact with different with materials. right? Materials are required to be able to deliver these processes. This is where corrosion becomes a very big problem. So if you take the reactor core, for instance, you're going to be exposed to irradiation, molten salt creep, high temperature creep, induced creep, and different salt. In different part of the heat exchanger loop, you're going to have flow-induced effect on your material degradation. And so you have all of these taking place. And also when you have pumps, you have valves, you have different kind of material degradation mechanism taking place. The challenge for us, of course, is to try and reduce the cost of thermal electricity, either from solar or from the molten salt reactor, and also to push the efficiency to above 50 degrees C. Currently, it's below that. And as I've mentioned earlier on, for you to be able to achieve this sort of efficiency, you need to be working your power block, power cycle at these temperature ranges. At the moment, the CSP, based on nitrate salt, cannot really achieve that, right? So you need a higher temperature. So you need new materials, or you need more resilient materials that can withstand corrosion. So that's the challenge. And again, we have to go back to the different salt chemistries. Now, I will mention that a lot of chemists are still working up different chemistries to improve the temperature ranges for which you can use these systems and generate your thermal electricity. So the most here is liquid lead based liquid metals, which has the highest temperature there. So that's kind of the molten salt. So the typical interface you would see is, you can kind of analyze it in this way. So you have your substrate, you have the molten salt, you have speciation within the chemistries, you have oxidation generating depending on the temperature and how stable the molten salts are. You have neutron irradiation for the MSR scenario. So I will share quickly some results that we've done in the labs, generated in the lab. So we did simulate some of these degradation mechanisms based on nitrate salt in the solar context. But we also try to do some work using Flanax salt, which is fluorides of sodium and potassium. I'll try to combine that with some numerical modeling approach. But I'm not going to present all of the results. I'm just going to focus on the one we did on the solar system, the CSP system. So this is a case in point from a collaborator in China. And they've had problems with nighttime cooling. They want to see the effect of nighttime cooling and daytime heating. And that's why we came up with an idea of having what we call thermocycling. So you can see the peak time is 12 hours during the day. Then you have another 12 hours of 290 degrees C. Now this temperature is just to ensure that your molten salt does not freeze within your receiver or through the pipes. So it's maintained at that level with some level of insulation. But then in some cases, extreme cases, there's a sudden change in the weather and then there's actual freezing. And they've come up with the time scale and the kind of temperature they've witnessed. So we kind of created what we call thermal shock within the system as well. So we've kind of tried to simulate that in the lab, but also tried to run some experiments where we vary the gas atmosphere to see the impact. Because part of the challenge we have with the CSPs is that no one knows, there are no data to show what is the chemistry of the atmosphere when you have this molten salt, because they are low vaporizing salts. Okay, but they have a high specific heat capacities. So, what have we got? So we have this. I think it's better to come. So we have this. I was just focused on these highlighted results. These are from weight loss measurement, from having the coupons of some different materials in nitrate salt. So we've created two atmospheres, one in argon and one in air at about 560 degrees C. And what we realized quickly was that based on normal convention, you expect when you have oxygen, you have a lot of corrosion, right? But what we realized is that when you have argon in the system, you have more material loss. And that was pretty much on because of the effect of the chemistries of the salt. So basically, the argon kind of catalyzes the transition from nitrate to nitrite and release of oxygen within the system. So there's no oxygen when we put in the salt, but by virtue of the processes taking place, you tend to have oxygen generated, and this oxygen leads to the formation of different oxide. Remember, we are dealing with stainless steel, so it's a multi-alloy system, right? So you have chromium oxide forming as an inner layer, and then you have iron oxide forming as an outer layer. And then we quickly realized the formation of a top layer, which is sodium ferrite. And this reaction takes place by the dissolution of this iron oxide. And what we have, we did some mechanical nano-indentation testing, and we realized that this outer layer, which is number one here, is just sodium ferrite, is very brittle. can easily spoil off the surface. And we quickly realized that you can go through a cycle of sodium ferrite breakdown, exposing the iron oxide to form more sodium ferrite. It breaks down, and then you end up losing materials. So this is what we realized from the results. So you can see the regions here. This is for 3, you have a lot of sodium-rich phases there. So we did a lot of analysis, XRD analysis of the salt chemistries before and after, and then we saw a lot of nitrite phases within the salt chemistry. So definitely confirming that the reason why you have a lot of corrosion in the argon atmosphere environment is because of the production of ferrite. Sodium nitrite in the system. So the sodium that is driving the formation of a more brittle sodium ferrite is from the salt, not from the material itself. So a lot of these, in terms of how material will behave in these systems, is dependent a lot on the salt chemistries. So we did a test in the molten salt reactor contest, which is for the nuclear. And what we had was the Flenac salt, right? But instead of having something that has to do with this, there's no oxygen, it's fluoride, but we started having chromium fluoride, part of the salt chemistry, as the agent of material degradation in that context. We also have hydrogen fluoride being formed as well. So these are quite aggressive to the materials when in use. So this was one of the key outcomes for this work, which was already published. And we also did the result from the different thermal cycle profile, eating profile. So we noticed the black line, which is the chart here, the points here. This is for thermocycling. The red one is for isothermal, and the blue one is for thermoshock. So we do we did expect we did expect the thermoshock to be higher, but what was really surprising was the thermocycling effect. The corrosion rate was lower over a period of time, and the reason we figured out was because you have the cooling effect. So you have 12 hours of cooling at night. What that tends to do is to suppress the kinetics of oxidation that you get with these reactions. So all these reactions that you get, especially the oxide formation, ferrite oxide formation, they are very dependent on temperature. So you've had 12 hours of cooling. So you've suppressed the kinetics of that process. And you can see that from this chart here. So this is with thermocycling. After 4 weeks, there was evidence of no formation or transition of your iron oxide to ferrite, sodium ferrite. After 4 weeks in isothermal, you can see the sodium ferrite showing evidence of spallation, but under thermal shock, there was no sodium ferrite available at the surface. And this is the reason why you have a lot more material loss in this system. So what we did conclude from this work is that the chemistry, temperature is important, chemistry is important, and just as I've shown in the previous slide, the speciation of the salt, the molten salt itself is quite important because you get a lot of secondary species that are formed, and then that translates into severe corrosion. And I think that's all I have time for today. Thank you for your attention.
Thank you very much. So now it's a question from the audience, please. Right. Anyone?
Thank you, Frederick. Just saw you have Inconel
yes.
in the list. Have you checked the chemistry on the surface? I mean, also I see the temperature range is around 750.
Yeah.
I mean, have you considered any kind of creep issues with stainless steel at this temperature range?
Yeah, so we didn't check creep, but we do suspect that creep is going to be, we didn't check creep because we think we're going to do a lot of subsurface analysis to be able to see a lot of creep. But we do recognize that creep is part of it. I think when we did the work in the MSR, we did at 750. This was at 560. At 750, what we did realize was, the modeling, we did some computational work. We modeled it by diffusion, and diffusion of the atoms, the chromium, nickel to the interface for it to interact with the salt. So we factored that into the approach we're doing. And that is because of understanding of the effect of creep as part of that. But we did not really do a lot of extensive investigation on that front. Yeah. but it's something that we are very aware of at that temperature. Yeah.
Right.
Yes.
Edmund?
So can I, not knowing this field at all,
Yeah.
you obviously posed some nice results in the system you're looking at.
Yeah.
You showed a whole range of different systems, and I'm not quite sure I understand what is driving you to a particular system to look at, and whether there are other systems that aren't on your chart you might look at. So could you say a little bit about which molten salt systems how you select the one to work on, and the sort of what other options there might be you haven't even looked at, if you see what I mean?
So what you said, just to kind of understand the question, when you said the molten salt, is it the context or just the salt chemistry itself? Because
I suppose I was looking at the chart you gave.
Oh, the chart. Okay, yeah.
You had a whole range of
Yeah,
materials.
yeah. Okay, all right. So
I'm not quite sure.
if I can go back, yeah. So there's a wide range available. It depends on this one, right?
That one. So lots and lots of things.
Yeah.
You pick
So
one, and I was just wondering.
So you have oils. These are all heat transfer fluids. Any of these can be used to kind of transfer the heat from the sun to the turbine for power generation. The motivation to use any one of them depends on what temperature you want to work on. So for instance, the most common one is solar salt. It's a nitrate salt, okay, for solar. And most of the highest temperature is about 600. You have 565. So these temperatures helps, it's more like you know how much heat you're going to be able to transfer by virtue of these temperature ranges within which they are stable. So most people use, I think there is a FLANAC. This is lithium, sodium, and potassium carbonate. A lot of them has to do with the chemistry. People think nitrate are more oxidizing. If you go at higher temperatures, they decide to use carbonate. Carbonate gives you more temperature ranges to work with, so you can get a lot more output in terms of how much heat you can conduct from the transfer from the solar system to your power block. So the decision to use this is your understanding of how this would affect the integrity of your materials. So the higher temperature you go, you have to decide what material you're going to use to build your pipelines, to build all the infrastructure, and see if they're going to be able to be resilient for a long time. So if you want to be more economical with your material choice, you have to work at the lower temperature ranges. So for most CSP, they use stainless steel. So because of that, they tend to use the nitrate salts. So you work at the lower temperature of 500 degrees C. But in the molten salt reactor, you're going to be working at 700 degrees C, 700, 750. You can't use nitrate. You're going to use what's it called, FLANAC salt, which is fluorides of potassium, nitrate, and sodium. But then you can't use stainless steel in that context because the material is going to degrade. So you have to decide on the material you're going to use. So understanding the interplay between material and how much heat you want to get out of the system or how efficient you want the system to be is very key on deciding which material to use or which sort of chemistry I'm going to use in these systems.
Okay, so it's very much a whole system you've got to look at in order to determine
And the push, one of the motivation is, I mean, we're talking about nuclear fusion now, so you're going to be generating higher temperature in the core of your reactors. That means you need to work at a higher temperature, you need more materials, you need to use more chemistries that can withstand those kind of temperature ranges.
Thank you. Yeah, I suppose for a complicated system with multiple material choices and components, so that's going to be a lot of things to consider, yeah, especially looking at high temperature corrosion or hot corrosion. So let's thank Frederick again for an interesting talk. And let's move on to our third speaker of this session. So we got Dr. Jamie Mannering from School of Chemistry at the University of Leeds. Jamie completed his PhD in Leeds with Robert Menzel and Rick Brightson, where he developed 3D porous nanocarbon and metallic targets for plasma physics experiments in collaboration with AWE. Jamie is now a postdoc with Robert's group, working on several projects related to integration of functional nanocarbon foams within catalytic flow applications. So he's going to talk to us about high entropy alloy nanocatalyst within ordered 3D graphene architectures. Thank you.
Thank you, and welcome. Today I'm going to talk briefly about one of the projects that I'm working on at the moment, and that is high entropy alloy nanocatalysts within ordered 3D graphene architectures. First of all, I'd like to spend a bit of time talking about the graphene architectures. These are the supports on which we deposit heterogeneous high-entropy alloy nanocatalysts. And for me, these are freestanding, interconnected, porous foams. And they have a unique combination of properties that are inaccessible within powders and films. So they have typically have a very high surface area, extensive open porosity, and as we'll see later, desirable electrothermal conductivity. And it's for these reasons that graphene foams have been utilized in a myriad of different applications, including sensors, energy storage, catalysis, and composites. And just to illustrate that point, I've plugged two recent publications down here on the left and right, where graphene foams have been utilized within battery research and within electrocatalysis. So forming these foams, typically through wet chemical approaches, it's a two-stage process. And this involves physical or covalent cross-linking of a nanocarbon building block. This can be graphene or carbon nanotubes, followed by the drying, because we need to remove the liquid, typically through lyophilization or critical point drying. And for me, throughout my PhD and current PDRA position, I've focused on trying to understand and control the assembly process to form a variety of different structures and material compositions. To just showcase that, throughout this time I've made some carbon nanotube microcages. This was published in Advanced Materials in 2021. Some stochastic carbon copper nanofibers, they're dendritic copper, as well as pure metallic capsules, and finally some lamellar graphene foams. And it's these lamellar graphene foams that I'll be focusing on for the next couple of slides, purely because they're conducive to flow applications, and that's something that we're quite interested in as a group. So the way in which we form these lamellar graphene foams is through a hydrothermal synthesis. Typically, we start with an oxidized graphene dispersion, stabilized through hydrogen bonding. We can deliberately destabilize that dispersion and encourage partial restacking through pi-pi interactions of the underlying graphitic substrate. And what this essentially does is form a perfect, templated by the overall container that's in, but it essentially forms a wet foam known as a hydrogel. And at this point, we can actually freeze the liquid within these foams to form that lamellar morphology. Now, there's a lot of flexibility here because we can freeze the water or the ice, the liquid within the hydrogels at several different temperatures to get different lamellar pore sizes and at different orientations that matter when you confine a liquid within flow. After we've done that, we need to remove the ice through lyophilization and then apply a final thermal reduction step to fully restore those desirable graphitic properties. So as I mentioned, these foams are electrothermally conducting, but because they're not perfect conductors and there is inherent resistivity there, what we can actually do is quite effectively convert electrical energy into thermal energy, and we can use that as a tool for really precise, rapid, and efficient thermochemical transformations on the surface of these foam structures. I'll show you a little video of that. So as we pass the current through, we have essentially black body emission. So these foams can go up to 3,300 Kelvin with extremely rapid heating and cooling kinetics. Now, a colleague of mine, Dong Shah, has looked at modeling high temperature gradients within these foams, and he's used that information to control the size of a variety of different monometallic nanoparticles. That, again, was recently published this month in JAC, so if anyone's interested, I'd encourage people to check that out. It's a really interesting paper. But for me, in my research, I'm actually interested in a different direction of exploiting these rapid kinetics to form multimetallic nanoparticles. So the reason why we want to work with multimetallic nanoparticles is that they often have different properties compared to their monometallic forms because of metal-metal interatomic interactions. This same line of reasoning can also be applied for the different configurations, atomic configurations of multimetallic nanoparticles. So to put this sort of all into context and provide a little bit more detail, if you try and mix 5 or more metals, under typical conventional heating and cooling kinetics, what we end up with is a thermodynamically driven, heterogeneous phase-separated structure. So all the different types of metals will separate into their own domains. But if you have a method or a tool of where you have rapid heating and cooling kinetics, you can end up really small, atomically mixed, crystalline nanoparticles. These are referred to as high entropy alloys simply because of the disorder within the atomic configuration. And to put that into a bigger picture, we can actually go one step further with even faster heating and cooling kinetics and form to the point where the metals do not have time to rearrange into a crystalline structure, and we end up with these amorphous metallic glass nanoparticles. But today, exploiting their electrothermal properties, we'll be focusing on these high entropy alloys. And for me, I wanted to functionalize the graphene foams with the high entropy alloy nanoparticles in a structure-preserving way, because if we have structure preservation, then we can have accurate structure function and structure performance relationships, and that has an impact across a wide range of applications within catalysis, within electronics, and within a wide range of other applications. So I've been working with Tom Chamberlain and one of his PhD students, Rebecca Stones, on using a sublimation deposition approach. We essentially, for this particular research as a proof of principle, I took 5 different metallic acetylacetonate and carbonyl precursors, and through a controlled phase transition, we can have these precursors in concerted formation, sublime, diffuse through the graphene foam, and then preferentially deposit onto the graphene foam itself whilst preserving the lamellar morphology. And then we just applied that electrical heating approach to rapidly decompose the precursors into the high entropy alloy nanoparticles. So I won't spend too long on this slide because it'd probably take more than 8 minutes to discuss this in detail, but we've worked with several different groups with Nicole and Zabida over in Lemus, and Sean Collins on aberration-corrected STEM at the SuperSTEM facility in Desbury. Well, what we can essentially see here is that we have all 5 metals that have successfully mixed in a homogeneous way. We have some high-angle annular dark field imaging, so scattering microscopy images through Z contrasting. And we can complement that with sort of understanding spatial frequencies along the columns of atoms within the high entropy alloy nanoparticles and comparing that to monometallic forms. And this complements the mixing and gives you a secondary approach to really understand whether we're actually homogeneously forming those multimetallic nanoparticles. And then we're also working with Sven Schrader and Andrew Britton on applying in situ thermal heating, XPS, to understand thermochemical transformations throughout different stages of heating. And we're starting to see differences in the composition and the electronic structure between the high entropy alloy and the monometallic forms. And we're sort of working through that now over the coming months to understand what's really going on there. In addition, we can look at the different profiles of heating as well. So we can do single pulse heating. So this is literally 1 second, 3 1-second pulses or continuous heating. And we can see by controlling this and looking at different profiles, we can form a new type of potential carbocatalyst by, so at the different heating parameters, we can control the thickness of a carbon layer around the high entropy alloy nanoparticles. This opens up a new opportunity because you can control the composition of the high entropy alloys that can have an effect on charge transfer effects to the carbon layer. And this just basically opens up the area in a new direction. Very briefly, I've included here some tomography data. So I've worked with a couple of people over at the Manchester National X-ray Imaging Facility, and we've got some X-ray computer tomography data here, and we've done some segmentation on this to understand network density. And this all has an impact when you start to put these foams into flow. Another aspect of the PDRA position that I'm currently in is essentially setting up a flow platform, which is a challenge in and of itself. And we have one set up now. I've tried to, I have put these graphene foams into a standard commercial OmniFit column. And we've tried to see how catalytically active these high entropy alloy nanoparticles are within a straightforward fine chemical catalytic reaction. And there's a lot going on here. It's quite a wide design space because there are different metals. How do those metals interact with one another? Actually, this lends itself quite a bit to automation and maybe a bit of machine learning, so a lot of this process could be streamlined. But very briefly, what we actually see is the high entropy alloy nanoparticles are indeed catalytically active. It shows a really good reactant conversion, a formation of the desired product, and a suppression of byproduct formation. And similar to the microscopy and spectroscopy data, we'll be spending the next couple of months trying to really understand what is going on there at a more fundamental level. This brings me to the end of the talk. Going forward, I'd like to see these materials in a wider range of applications, particularly within CO2 utilization, perhaps with value-added C3 plus chemicals. CN bond formation using waste reactants, perhaps within graphene research, maybe pushing the boundaries on the material aspect as well to form metallic glass nanoparticles, and then exploring these within a wide range of flow reactions, as well as looking at them with using the electrothermal heating for single and dual atomic site catalysts as well. I'd like to thank all these people that have contributed to this research, in particular the PI, the PI, Robert, Sean, Rick, Nicole, and I don't know why his name's so far down, but Dong Shah has really laid the foundations for a lot of this research, and I really need to thank him for that, as well as these funding facilities and institutions. Thank you.
Thank you very much. Very nice talk. So, questions from the audience, please. Rick. Yeah.
I mean, are there examples of high entropy alloy particles where the individual constituents aren't all catalytically active, if you see what I mean? Because I noticed you were using things like platinum, ruthenium. I just wondered if there was any weird effects, you know, that...
No, it's an interesting question. A lot of the metals in the literature, certainly, where these high entropy alloy nanoparticles have been formed, are always with sort of the early transition metals. Quite a lot of those are active in a lot of different reactions. If you're referring to a particular type of reaction, you could probably think of a way in which we could couple some alkaline earth metals, maybe. They're less active in a particular in a given reaction, but I've not really seen a lot of work done on actually considering that aspect of high entropy alloy formation, and it's something that we can consider in the future.
Might lead to sort of more sustainable catalysts, maybe, if there were examples.
Any other questions? Right, so I think this has come to our close of the session, and we'll have a comfort break before our next session. Thank you very much for all the speakers in this session, and we'll see you shortly. Andy, are we back at 3 or 11?
Yes, well, we're running about 10 minutes over, so I think if we're 5-2 now, so if we start again at 5 minutes past 3.
This next session. I'm very pleased to welcome our first guest speaker, Lorenzo Di Michele from the University of Cambridge. Lorenzo spent most of his career at Cambridge with a brief stint at Imperial College and then went back to Cambridge, quite right too. And he's held a number of very prestigious fellowships from the Lever-Hughes, from the Royal Society, URF, amongst others. He's got an extremely impressive track record in DNA, nanotechnology, soft matter, bottom-up synthetic biology, and so on. Today we're going to hear about some of his work on synthetic cells for smart nucleic acid condensates.
Thanks a lot for the super kind introduction. It's been great to be here today, seeing so much work on all sorts of aspects of materials. So we are going to change topic. Oh, sorry.
Hello? Great. Start again. So thanks a lot for the very kind introduction and for the great talks to everyone who has already spoken. So yeah, we're going to change topic completely from the metals and corrosions and nanoparticles that we have seen in the previous session to soft, hopefully smart materials made out of DNA, trying to chase this slightly crazy idea of using them to build things that look and behave like biological cells. So I'm not sure how many of you have encountered this concept before, but the idea here is that we want to build synthetic cells. And these are devices, micro devices, typically constructed completely from the bottom up. So starting from non-living molecular components, things like lipids, enzymes, if you want, nanoparticles, nucleic acids, and so on. And then we want to put them together, hopefully in a rational way, trying to build these synthetic cellular devices. And the idea is that, of course, you don't want to build something which is exactly like a cell, but rather you want to build a device that can do some things that people might kind of connect to what a cell does. And then depending on what your application is, you might focus on one or the other. For example, one might want to build synthetic cells that can do sensing, for example, to sense an analyte associated to disease, or that can produce interesting, useful molecules like nucleic acids or peptides that can communicate with each other or with live cells that can move in a specific way, for example, doing chemotaxis, and perhaps in the future that can grow, divide, and maybe one day evolve as well. There's many good reasons for which one might want to try to solve this quite tricky problem. One of them is that these synthetic cells make excellent models for studying biology because, of course, you can recapitulate a complex biological phenomena in a much simplified systems in which you only put the components that you need in order to observe the phenomenon that you're trying to study. And like that, you can learn about it. There's many groups who are using synthetic cells as tools to study the origin of life. In this case, you don't quite have the complete flexibility in choosing your building blocks and your components because you have to make them realistic, given the chemistry that existed back there. But still, they make extremely useful models. But there's also loads of potential applications for these devices. Many people, including us, are trying to design synthetic cell devices to be used in healthcare, for example, as synthetic cell therapies to carry and deliver vaccines for diagnostics, for example, cancer diagnostics, and for bioprocessing. Now you can make synthetic cells really with anything that you might kind of be an expert on, really. So there's people who focus more on the lipids, there's people who focus more on the transcription and translation machinery, or the enzymes, or the membrane proteins. And the angle that we try to explore in our group is to use the tools of nucleic acid nanotechnology. I know that there's some people in the room who are experts on that, but maybe many others we find this still quite new. It's actually quite an old concept now that was introduced by Professor Ned Seaman, who sadly passed away just a couple of years ago in the early 80s. And the basic idea here is that you want to use DNA not just for its kind of role as the molecule that carries genetic information, but you want to use it as a material, as a self-assembly material. And you can do so because of the very strong selectivity of base pairing. So if you design 5 DNA molecules such that they have base pairing interactions in a specific way, you can design exactly how these molecules will come together and will self-assemble into a specific nanostructure. And you can do this with not just 5 molecules, you can do this with thousands of molecules because you can have loads of different sequences. And you can synthesize this very easily, or you can pay a company to do it for you, and then you can self-assemble devices and nanostructures with really all sorts of shapes, from the very simple objects like these tiles, these junctions, all the way to things that are quite large and extremely complex, that typically would make with the technique known as DNA origami. And what we are trying to do really is to use DNA nanostructures and also RNA nanostructures, as we are going to look very briefly at the end of this talk, to control and program both structural aspects as well as functions and information processing in synthetic cells. We have a number of projects on this. For example, we have kind of some research lines looking at engineering the membranes of synthetic cells using DNA nanostructures attached to them, in which we try to make membrane-less compartments out of DNA for our synthetic cells, and we're going to expand on this quite a bit, in which we try to engineer interesting behaviors, for example, communication with live cells and division-like behaviors and motion. And then we have some projects on applications, in particular to antimicrobials and cancer diagnostics. Of course, we're not the only ones doing this. There's many groups all around the world trying to, this is slightly kind of weird, it has shifted, but there's lots of groups trying to tackle this problem around the world, and here I just quote a few of them in Germany, in Japan, and in Los Angeles for kind of geographical diversity. And you can read more about these, some recent results on using DNA nanotechnology in the context of synthetic cells in this very recent review. But okay, so let's now start with the proper results. So let's see how we can go about designing and building a synthetic cell, primarily using DNA nanotechnology. And we're going to do this in steps. So we're going to start from the simple and then making them a little bit more complex as we go. So the first ingredient that we need to build a synthetic cell is a scaffold or a compartment. So this will contain all of the functional machinery and somewhat separate the inside from the outside of our synthetic cell. But then we would need to make this compartment a little bit more complex. So we would have to be able to establish within it some addressable regions in which we can localize specific functions. So we can regulate what goes in there and what does not go in there. So basically something that plays the role of the organelles in our biological cells. And then finally, we need to kind of take this cell-like device and give it some interesting cell-like functions. And as I said, there can be many of these. We're going to just focus on one today because we don't have a lot of time. But let's start from number one, thinking about our scaffold for the synthetic cells. So in many cases, you will find that people use membranes to create the containers for synthetic cells. This is because it imitates the membranes of the cells. In most cases, you would use lipid membranes, but you can use other types of membranes as well. For example, polymer membranes can be good because they are quite robust. And as I said, we do a bit of work on this too, and here are a couple of references by Roger, who's a research fellow associated with the group. But today I want to talk about a different approach to creating compartmentalization in synthetic cells, which doesn't use membranes. It uses membrane-less compartments. And this is, of course, inspired by the fact that people have recently realized how membraneless organelles are quite ubiquitous in cells. They use them to dynamically control loads of processes. And we can generate similar compartments synthetically, for example, droplets, coacervates, condensates, synthetic biomolecular condensates, and hydrogels. They have advantages. For example, they are much more robust, typically, than your membrane-based compartments. They also have the advantage that you can make them out of DNA, which for us is a big advantage. So let's see how we can do that. You can do this using different types of DNA nanostructures, but the one that we have developed over the years and we still use a lot are these very simple building blocks, which are DNA junctions, right? So they are quite flexible here in the middle, and this is just duplex DNA. But these are a little bit different from similar objects that you might find in literature because they are functionalized to have a cholesterol molecule attached at the end of each little DNA arm, which makes these building blocks amphiphilic and gives them quite interesting self-assembly properties. So these assemble from a small number of distinct DNA molecules, some of which will be functionalized with cholesterol and others are not functionalized. As per well-established tradition in DNA nanotech, all that you need to do is to mix all of these molecules together, heat up, cool down slowly, and these molecules will self-assemble to form these little crosses, and then these will also assemble, driven by the hydrophobic forces between the cholesterols, to form a network-like material in which you have all of the cholesterol molecules coming together in little hydrophobic pockets, and then this will be cross-linked in 3 dimensions by these DNA junctions. Depending on the exact design, on the buffer conditions, and on the rate at which you quench, you can obtain materials which are very obviously crystalline solids. As you can see, you have large single crystals, or you can obtain droplets. And these can either be gel-like or can be liquid, depending on temperature, again, depending on salt concentration and design specifics. We're going to refer to all of these as condensates, just for simplicity. For the case of the crystals, we have done lots of characterization. We have taken them to diamond and done small angular X-ray scattering to figure out that they have a body center cubic unit cell, and we can measure the lattice parameter of it. And importantly, we can control very precisely the lattice parameter by changing the length of this DNA arm. If you make it longer, the lattice parameter increases linearly with it. And this is very important because by changing the lattice parameter, we can change how porous these frameworks are. And it turns out that if you make the lattice parameter large enough, the pores are sufficiently large for things like proteins and enzymes to diffuse in. So in this case, you can see that things like streptavidin or GFP can diffuse inside these aggregates. And this is going to be important later on to give these objects some interesting functionalities. We can also induce structural responses, the simplest of which is a disassembly reaction that we can induce by adding a nucleic acid trigger, which is this molecule here that through a process called a strand displacement reaction binds to another DNA molecule, which is a structural element in these stars, and peels it away. And this connection that we call a bridge molecule links the center of the star with the cholesterol molecule, with the sticky bit. So once you remove it, everything falls apart, and you can see this in the video here. We can also have responsiveness to other types of stimuli. So for example, here we have achieved response to the presence and concentration of potassium by replacing the central junction of our star with a tetramolecular G quadruplex. So this is a non-canonical DNA nanostructure that is strongly stabilized by the presence of monovalent cations, in particular potassium. So when you don't have potassium, this little connection is not formed, so the star is just not there, and you have no condensates. When you add the potassium, this little knot forms, and then the stars form, and then the condensates start forming, and you can dissolve them very quickly by chelating away the potassium using a specific chelating agent. So this was a very quick overview over a few years of research conducted by some very brilliant PhD students and postdocs, faces shown there. But it's really just to convince you that these kind of condensates, synthetic condensates made of amphiphilic DNA nanostructures could be potentially good materials for creating scaffolds for our synthetic cells, because they have loads of desirable properties. We can make them about the size of a cell. We can actually control the size. They are very stable. They're very robust. We can make them very easily. We can make them responsive to lots of different stimuli. So I've shown responsiveness to presence of certain nucleic acid molecules, to salt, but you can easily make them pH responsive or light responsive as well. And importantly, we can regulate diffusive transport in and out of them. So let's say that we are happy with our basic scaffold. Now, the next step is to make our scaffold a bit more interesting, and in particular, to create chemically addressable compartments within it, within which we can then localize specific functions, a little bit like it happens in a eukaryotic cell. So we worked on this problem for a few years, and we came up with two alternative solutions, the first of which was the subject of the PhD of Adrian, who recently finished, and uses reaction diffusion processes to pattern our DNA condensates. And then the other approach, which has been recently published by current PhD student Leila, instead uses phase separation. And briefly, we are going to look at both of these strategies. So once again, our objective now is to take a uniform condensate and make it non-uniform, so to localize specific molecules in specific subcompartments. For the case of the reaction diffusion approach, all that we need to do is to modify our elementary building block to attach this binding site, which is a longish DNA molecule, and then consider patterning strands which can bind to this binding site. These DNA strands, in this example, we have two of them. We have this short one that only binds to this little bit of the DNA of the same color, labeled with a red fluorophore. And then we have a long one, which binds to the whole of the binding site and is labeled with a green fluorophore. We generate our DNA spheres, and all of the little nanostructures that compose them have this binding site. Then we add these patterning strands on the outside, and because now this sphere is porous, they will diffuse in. Now, the small molecule will diffuse in much faster because it's a porous environment, so there's kind of big difference in diffusion constant with size. So the red molecule goes in first, and it binds to the free binding sites. So you get this red front. But then if you wait a bit longer, also the green molecule will start diffusing in. Does so at a much slower rate, but it does. But what it does, it will actually displace away the red molecule, because the green molecule can bind to the whole of the binding site. And entropically, you never have two molecules connected. That would cost kind of extra entropy for localizing two molecules when you gain the same in enthalpy, because the green molecule can actually occupy the whole thing. So the red molecules are released back into solution. And you get this green wave that progressively consumes the red, until at the end, you only have green. But now, at these intermediate times, we have basically achieved what we wanted. So we have now two different regions, in this case concentric, within our condensates, one that only has the red molecules and one that only has the green molecules. Now, these molecules are not particularly useful because they just have a fluorophore attached to them, but you can imagine how you can then use this to connect other components which can bring function and localize function in specific pockets of our synthetic cells. So this is just a video that should be going, that shows exactly what I described. So you have the red going in first, followed by the green, and then everything becomes green at the end. You can play the same trick with more than two molecules. So here we have a third molecule, which is non-fluorescent, and it's intermediate in length between the red and the green. So what it does is that it generates this black band between the two colors, which now is not empty, but it's where this third molecule is. So here, after 12 minutes, you have 3 chemically addressable regions within your condensate. You can have 5 if you want to kind of push it, and then you've got lots of rings that quickly evolve. But if you look after 7 minutes and you do some image analysis, you will find that there's now 5 concentric rings. And the maximum we could do, just to really stretch it, is 7 at the same time. Got lots of things that move, but again, if you look at this after 30 minutes and you count the dark and the bright rings, you can convince yourself that you have 7 distinct regions. And that's really as far as you can push it, because then the patterning strands become close to each other in terms of size and binding strength, so they don't separate well anymore. Of course, this is kind of rather useless if you don't have a way to arrest it and freeze it, because if you let it equilibrate, eventually the strongest binding molecule will kind of uniformly occupy everything. But it's very easy to freeze these reactions in place. All that you need to do is to add an excess of what we call a stop strand, which has the same sequence of this binding site. You add it in solution and will capture all of the excess patterning strands. And when you do so, everything arrests. And this is because these fronts propagate only if there is a displacement reaction. So if there is a new molecule coming in and displacing a molecule which is already connected to a binding site. Thermally, a molecule on its own will not come off. So once you remove all of the molecules that can do the displacing, nothing evolves anymore, and these patterns remain frozen for several hours or as far as we have looked at them. In this video that just played, we added the patterning strand after 16 minutes, and you can see in the sequence of images that the position of the fronts simply stops evolving. It's just a little bit of photo bleaching because we keep imaging. So this is one strategy to achieve this kind of patterning of the DNA condensates, but there's another strategy that very, very briefly I'm going to touch on that uses now phase separation. So here the trick is that instead of having these aggregates forming from just one type of DNA star, we're going to use two different designs that have different size or different length of this DNA arm. So we call A the short one and B the long one. We assemble them together, and as you might expect, you get phase-separated core shell geometries in which the inner phase has a tendency of being enriched in the small structures, and the outer phase has a tendency of being enriched in the larger structures. We have done lots of characterization on this that you can read in this paper. We tried lots of different combinations of stars with different designs and different size, and it turns out that the driver for this phase separation is simply the difference in size. Indeed, if you look at the kind of likelihood that a given aggregate will display phase separation, you have this sharp transition where between a regime in which almost none of them is phase separated to a regime in which most of them are phase separated as a function of the ratio between the length of the arms in the large stars and the small stars at around 1.2. And we have looked at the structure of these phases, and you have sometimes coexistence of a crystal phase with an amorphous phase. Sometimes you have two crystals, and you can read about it down here. But this was just to give you another example in which you can generate heterogeneity within these synthetic cells. So let's say that we are now happy with our second step. So we have started from a kind of simple, uniform aggregate, and now we have made something which has internally addressable regions within which we can localize certain functions. So our final task is to think about one of these functions and localize it within our synthetic cell. And one of the simplest things that you can do when you have DNA around is try to transcribe it to make some RNA. That's what actual cells do. So that's what we wanted to do as well. So in this case, we use the reaction diffusion approach to pattern otherwise uniform condensates and localize in the center of them now a quite long bit of DNA, which has a promoter region that can be recognized by a RNA polymerase called T7 RNA polymerase, and then codes for a functional RNA nanostructure. So the polymerase can read this bit of DNA and make some RNA. And this RNA nanostructure is called the fluorescent light-up aptamer. In particular, it's called broccoli, because it can form a complex with this otherwise very bad fluorophore that when it binds to a certain pocket within this aptamer, increases its quantum yield significantly, and so you see that you've made loads of RNA through fluorescence. Then we have modified our broccoli aptamer to have this little handle, and we have localized in the outer ring of our synthetic cell binding sites for this handle. And here the idea is that we have now two regions, a region in the center where the RNA is being made, and this will be what's equivalent to the nucleus of our synthetic cell, and then we have a region on the outside where RNA is being stored. It diffuses there and then it accumulates there, and this is something like the cytoplasm of our synthetic cell. After you've done the patterning, you add your RNA polymerase, as well as all the other bits and pieces that you need to do the in vitro transcription, and this will diffuse inside, and then RNA will start being made, and then the RNA will diffuse out. Let's see this happening. So in this video, the cyan spot is the nucleus of the synthetic cell. And when I click, you will see in orange the RNA accumulating. And you can see that it does so starting from this interface going towards the outside. And that's because it's being made actually within the synthetic cell and then progressively diffuses out and binds to the dedicated binding sites. And you can see this in this sequence of images as well. You've got from the inside towards the outside, the RNA accumulating. And importantly here, we are not actually growing new material. So that if you look in bright field, the condensate remains always the same size. We're just accumulating these small molecules in dedicated binding sites. So this takes us to the end of the main part of the talk, and we have gone from, again, relatively simple self-assembled materials. We have made them a little bit more cell-like, and then finally we have given them an interesting cell-like function, which is the ability to synthesize nucleic acids. And this, of course, could be potentially useful if you wanted to build a synthetic cell that produces a therapeutic nucleic acid, for example, for gene therapy. So in the last couple of minutes, I really wanted to very quickly show a couple of highlights on other parallel or kind of follow-up research lines in which we really tried to push the functionalities of the synthetic cells a bit further. So in this work down here, we wanted to, which I'm not really going to describe the work itself, just the idea of it. We wanted to try to do something quite ambitious, which is build a synthetic cell system that imitates a complex response of the immune system, and in particular, something called netosis, which is something that immune cells called neutrophils do in response to large pathogens. When they can't kind of kill the pathogens by eating them, instead they do something quite dramatic and drastic, which is exploding and releasing all of their DNA, which forms a little sticky cloud of DNA and DNA binding proteins that wraps around these pathogens, immobilizing them and then killing them, because these DNA binding proteins are actually cytotoxic. And you can see this happening in this video, where these filaments are actually fungal filaments, and this little color cloud, these are the neutrophils, and the little colored cloud is this DNA. And this is called, again, netosis, and the DNA cloud is called a neutrophil extracellular trap. So we wanted to do something similar with our synthetic cells. Again, I'm not going to tell you how this is done, only that we managed to do something vaguely similar by using a consortium of two types of synthetic cells, some DNA-based ones, very similar to the ones that I've described, that work together with some vesicles loaded with antibiotics, and then together they can detect the presence of bacteria, in particular the acidification in the medium induced by bacterial growth. When they do, they can kind of form a sticky DNA network that traps the bacteria, and then the antibiotic is released and it kills the bacteria. And again, you can read about this down there. And then very, very last thing, very quickly, we've seen earlier that in the case of the RNA producing synthetic cells, we were kind of producing new materials, but we're not able to actually grow the synthetic cell. So we wanted to do that, and a way in which that could be achieved is by producing RNA and using RNA as a way of creating structure for your synthetic cell. So we designed some RNA stars that are very similar to the DNA stars that we have seen earlier, but now they are formed from a single strand of RNA that folds on itself in this star shape. And then these little stars can interact with each other using these loops that are called kissing loops that exist in biology. For example, these are very similar to the ones that you find in HIP. But now, because this is an RNA device, you can just take a DNA template that codes for it, and then you can transcribe it many, many times using your RNA polymerase and make loads of these. And these will then form condensates, and these will grow over time. And you can see this happening in this video. You have liquid droplets that grow, and they coalesce, and they become quite large. And then I will skip loads of this, but you can do this in binary systems of two different condensates that don't really interact with each other, so you get this phase separation as well. You can do this within synthetic cells, so you can have synthetic cells that produce their own RNA organelles over time, and you can control the shape as well of them if you have two phases. And we have a way actually to control the affinity between these two phases, but by having a linker star that mediates interactions between them. With this, I'm going to stop by thanking everyone who contributed to this work. So I've shown some faces already and the names of the important people. Well, everyone is important. The name of the people who actually did the work I presented are in bold here, and then some funding, and finally you for your kind attention.
Great, thanks so much, Lorenzo. Oh, we've got a question already there. Oh, yeah, it would be.
I just wanted to see Andy run, actually. No, thanks, Lorenzo. That was a really nice, interesting talk. I've got two hopefully very quick questions. One is, have you tried mixing liposomes with your cholesterol condensates? And does that give you a kind of mixed compartmentalized condensate, or does it just kill the condensation reaction? And the second one is, have you ever tried doing different length arms in your DNA stars, and does that kind of inhibit the crystal phase and just give you more liquid-like condensates?
Thanks for the two great questions, Paul. On the liposomes, yes. So it's in this. No, it's ahead. Sorry. So in this, I think I passed it. Sorry about that. Let me find the right slide. Yeah, this one. So in this thing here, we actually look at that. So basically, what we have done is to take these condensates and then add SUVs. And what they form is a layer of SUVs around the condensates. And it's quite interesting. They don't go in, obviously, the size that we have tried. They form this layer. But this layer is somewhat connected, because if you then dissolve the DNA condensates from within this layer of SUVs, the layer of SUVs forms a shell that remains there. But it's not a continuous bilayer, so they don't restructure into, say, a continuous GUV-like thing. They remain a collection of SUVs kind of linked to each other, but they do not disrupt the DNA condensate. If you put an excess of them, the ones that don't go to the condensate, they just remain in solution. Then your second question was on what happens when you change the size of the star. This is, actually there's a plot on that, which is this plot. So here what we have are kind of powder diffraction patterns obtained with SACs as a function of the arm length. And what you said is exactly what happens. So if you increase the arm length, you get crystal, 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 crystal. If you make the arm length too long, at some point you no longer form crystals anymore. You start forming kind of some amorphous thing, which is quite liquidy, depending on temperature. Arms of different sizes? So it's not symmetric? No. We have never tried that.
Good. Are there any more questions? Oh, yes, please.
I think you might have just mentioned it right at the end, but have you tried different lipids to try and induce your phase separation as well? Because obviously you've used cholesterol throughout most of it. I think you might have done that right at the end.
I haven't talked about that. So we have tried with tocopherol. We have never tried to have stars that are identical, but other than having cholesterol versus tocopherol. That we have never tried. We have tried stars with different size, one with tocopherol, one with cholesterol, and it works slightly different than when you have both of them with cholesterol. But this is something we are doing at the moment. But for example, if you use tocopherol, it largely behaves similarly in a single component system. So it forms the same crystal phase if the cholesterol would also form a crystal phase in that situation with a similar lattice parameter. But we should try just identical stars, one with tocopherol, one with cholesterol, and see what happens. We haven't tried that.
One final question, maybe we do. Jim. Oh. Perhaps you could all line up in order to ask your questions and save Andy.
So I have a question with two parts. The first one is just to make sure that I understand what is the structure of the vesicle wall? It's a crystalline vesicle wall?
So this condensates?
Yeah. I mean, I'll call it a vesicle. I'm assuming it's open on the inside and has a wall, right?
No, it's full. It's a three-dimensional material. So it's So it's like a network of these DNA stars, which are kind of all connected to each other.
It's a DNA crystal.
It's a DNA crystal, yeah. It's a DNA cholesterol crystal.
Okay, so that takes me to my second question. In biological systems, you know, obviously we don't know, it's not like biology had a philosophy and used lipids for a membrane and proteins for machines and DNA for, you know, blueprints, but that happened for certain reasons. And one of the aspects of lipids, of course, is that they're inherently dynamic as a membrane, so things can insert into them and come out of them. And proteins are conformationally dynamic, whereas DNA, you melt it and you freeze it. And it's like you know exactly what you're going to get. You can program. That's why it's so beautiful. Unfortunately, you know exactly what you're going to get, and you can't change that. It's not dynamic. So you're starting with something which is inherently, you know, crystal-like. So how do you overcome the lack of dynamism and reach the versatility that you get with lipids for a membrane and proteins for machines?
So obviously, great question. Will probably take like half an hour of discussion. So I'm going to try to be very brief. So obviously, I agree with all that you said. So the beauty of DNA is that it is programmable. So whereas until very recently, you couldn't do almost anything with proteins in a kind of designer fashion. Now you start to kind of be able to do that, but until very recently, you could not. So yeah, in the future, when we'll be able to kind of engineer protein systems similar to what we can do with DNA, it would make sense to do that. In terms of the dynamics, but in general, in terms of the kind of functionality, with DNA nanotech, we can do things, and we do things that don't actually happen in biology. So for example, you can kind of induce all sorts of kind of strand displacement reactions that kind of use the knowledge and understanding that we have of thermodynamics to induce, when we want, unbinding of something and binding of something else. And we can control the rate of this quite precisely. And this is typically how you would obtain dynamic responses in DNA nanosystems. But also the other important thing is that we can use chemistry to modify the DNA in ways that in biology doesn't exist. So, yeah, for example, you can attach kind of fluorophores to it, you can attach nanoparticles, you can conjugate it to surfaces, you can attach photoresponsive linkers or pH-responsive linkers. So we have a wider toolkit at our disposal.
I think Andy's approaching with menace. So let's thank Lorenzo again for a fantastic talk. Thank you. So our next speaker has just dropped everything on the floor.
That was great.
Our next speaker is Lorna Dougan, Professor of Physics here at the University of Leeds at the School of Physics and Astronomy. I think Lorna was a bit apprehensive that I might sort of read out a massive biography of her, so I'm not going to. We're going to hear about her work on translating mechanical properties of proteins across the length scales.
There we go. Thank you, Giles, and thank you to the organisers for this invitation to share our work. I'm very happy to be here. So I'd like to set the scene for this short talk by thinking about polymer networks. Polymer networks are ubiquitous in materials research. So polymer networks are an important class of soft matter system. They're made up of polymer chains which are interconnected in some way. And you can see some of the different ways that polymers can be connected for many different systems. So we can have these interpenetrating polymer networks. We can have slidable crosslinks. We can have physical and chemical interactions. And what is really nice about these systems is that the structure and the mechanics can be modulated by a number of different properties, including their chain length, branching, and molecular composition. And we've already heard some really nice examples in this meeting of the use of polymer networks. So let's consider some of the applications. And again, because this is a broad audience, I'd like to set the scene for some diverse examples. So first of all, polymer networks are really crucial for structural vulcanized or hardened rubber tires. And in this case, the polymer network structure adapts under the strain that it experiences during its function. And that network is really important for its dynamics and properties such as how the network responds to strain and how it fractures. In the second example, polymer networks are important as active materials for gas separation. So some of the considerations are the cross-linking of the polymers in the network and the entanglement of those polymer chains. And that can allow you to control the diffusion of gases through those networks. And there's a lot of activity at the minute in terms of using these networks for carbon capture and storage. And then in the final example, this is modeled by my daughter, who had a little fall. And if this has ever happened to you and there's been blood involved, thankfully in her case, a little scab has formed quite quickly. So this is related to blood clotting, which is a really essential biological process. And blood clots are self-assembled active materials. And in this little cartoon, you can see a depiction of bleeding occurring, and you can see the red blood cells and the platelets. If things go to plan, if things are functioning normally, then a really important part of this process is a polymer network, a fibrin mesh. Fibrin is a long, sticky protein. It starts to form a hierarchical structure, and that structure gives you some mechanical integrity. And so that plug, that clot, is capable of withstanding the forces that are induced by blood flow. And the mechanics of that blood clot are really essential for the function. So it needs to be soft enough that it can do its job, but not stiff enough that you can start to have some really serious disorders, such as in thrombosis. So that's just some examples of where polymer networks are important in materials research, but there are some really big challenges remaining for everyone working in this area. And I'd like to highlight two that are particularly important for my group. So the first one is, we understand a lot about how a polymer or how a biopolymer responds to mechanical forces. And there's been a huge advancement in terms of experimental and modeling techniques that have allowed us to think about the force response of polymers. But what continues to be really, really difficult is to construct a theory that translates the mechanics of that polymer to a network of connected polymers. So how can we do that? How can we begin to understand those theories? And we need those theories because we need to have predictive control over the polymer networks that we're forming. So that was identified as one of the major challenges of soft matter physics. That was back in 2014. And then more recently, last week in fact, this paper came out again highlighting this as a major problem for the field, and also highlighting that this is even more complicated because of the dynamics, the dynamic mechanical properties of these systems. They are relaxing, they are responding to their environments. So not only do we need to know how to translate them as mechanical forces, we need to understand that dynamic process and how it's changing with time. So those are the things that we're interested in. And to do that, all we need to do is uncover the multi-scale design rules of these systems. We need to understand their structure and their mechanics across the length scales. And in our case, the relevant length scales are going from the nanoscale through to the macroscale of the materials that we're trying to make. So again, this is a short talk. So what I'd like to do is introduce the technique that we're using to address these challenges and share some recent examples of the progress that we're making. So the approach that we take is to make protein-based networks or protein-based hydrogels. And in this cartoon, this dynamic cartoon, you can see the representation of folded proteins which are photoactivated and chemically cross-linked to form a percolating network in the form of a hydrogel. So a hydrogel is a polymeric-based material which is water-rich. Our building block, as I said, are these globular folded proteins. They have a well-defined folded structure. That structure is known for the proteins that we're using, and that structure is completely connected to its function. So in the first session this morning, and I completely agree, George introduced proteins as nature's bio-nanomachines. These are the things that do all of the jobs that we want them to do. So they catalyze reactions, they fight infections, they promote growth. So we've got this fantastic functional building block to work with. But not only that, the mechanical properties of these folded proteins, they are mechanically robust, and they are tunable, and the information that we have about their mechanics is now incredibly rich because of the explosion of information that we've had from the single molecule community, both experimentally and modeling. And the other big step forward that we've had, particularly in the last 5 to 10 years, is our ability to use protein engineering to create synthetic protein building blocks for these studies. And what I mean by that is we can take a folded protein, here it's depicted as a sphere, and we can connect it into a polymeric chain. So now we have a polyprotein chain of a very defined length, and we can control the way that protein cross-links to form the network by using mutations, for example. So we can control the density of the cross-links in the network that we're forming. So that's our building block, and what we're hoping to do is understand this black box in the middle, understand what's happening with the forces and the structure as it scales, these length scales. And if we can do that, we've got a really, I think, exciting platform that we can use to create these future biomaterials. And the properties of those materials include having programmable structure and mechanics. We want to exploit the dynamic properties of this system. We've got the inherent and diverse biological functionality of our building block. And crucially, these proteins are responsive not just to mechanical cues, but biochemical cues that they experience in vivo. So very briefly, how do we make them? So we start off with folded monomeric proteins in solution. We use a photoactivated chemical cross-linking reaction to trigger nucleation. And then in time, we have percolation across the system to form a spanning network or a hydrogel. We can use techniques such as rheology to measure the mechanical properties of these systems. So this is one of the rheometers that we use over in physics, over in the Bragg building. And we've just built a little device that allows us to trigger the photoactivation, in this case with a blue LED light source. And so you can see here, we can monitor the mechanics of the system, here measured by the storage modulus. We start off with our pre-gel solution, and then we switch on the lamp, and then we can monitor as the proteins start to diffuse and find each other in solution. They start to form cross-links, and with time, the network grows, and we have a gel forming, and then we switch off the light source, and we have relaxation. And so there's two really interesting parts to this. This approach of doing in situ network formation is allowing us to study network formation and network relaxation. So the dynamic evolution of this network with time under certain conditions. So let's think about what that might mean. So for any soft matter-based system, you can start to think about particles coming together, finding themselves, aggregating, percolating, and so on. And one framework with which to think about that is in terms of diffusion and reaction-limited aggregation, which we heard a little bit about in the previous talk from Lorenzo. And what's interesting about this is if you have a system which is diffusing around in solution until it finds something that it can cross-link with, and in our case, it's an irreversible chemical cross-link, then with time, a network will form, and depending on whether it's diffusion limited or reaction limited, a very different structure will form. And this has been demonstrated for many different systems over the years, and indeed, it turns out that it's extremely advantageous for our protein network systems. So in our case, we can control the reaction rate very simply through the intensity of the lamp in our photoactivation. So we can either drive it towards being a fast reaction and diffusion limited, or the converse, being reaction limited. And so over the years, we've been using rheology and small angle neutron scattering to look at the structure of these systems as they form through these different processes. And you can really get very distinct structures forming. In both cases, you have this heterogeneous network, and we've got these clusters of folded protein which are interconnected by unfolded protein. And if we go through this route, we can get larger clusters, we can get a more homogeneous structure that's not as dominated by this intercluster region. And interestingly, we get stronger gels. So this is really interesting, and it's providing us with a route to really control and modulate the gels that are forming. And we also use some coarse-grained modeling approaches to look at this network formation. And this is an example where I was going to say PhD student, now postdoc, Kalila Cook, has been using a lattice-based modeling approach to think about the diffusion of these proteins in solution, forming cross-links, and percolating to form this network. And we can really start to map out the design space of these systems with these coarse-grained modeling approaches. So I wanted to give you an example, because right at the start, I talked about the challenge of translating mechanics across length scales. So in this example, we're taking, oh, where's it gone? That's strange. Here it is. So this is a representation of a protein called maltose binding protein. It binds specifically to maltose. And using AFM, not in imaging mode like we heard about in the first session, but in force spectroscopy mode, we can mechanically manipulate these proteins, and we can measure what force is needed to unfold the protein. And what was shown a number of years ago is that when this protein is bound to maltose, so when this little Pac-Man has eaten something, the mechanical stability of that single protein increases. So an interesting question is, if we use this system as a building block to form a network, do we see that translation of mechanics? So in the way that I just described previously, we make a cross-linked protein network, and sorry, it's coming up differently on my screen. Yes, we use rheology to measure the storage modulus of the protein network. And what you can see that in terms of this storage modulus, we see an increase in the presence of maltose. We can combine this with modeling to extract an internal force that's needed to unfold a protein in these systems. And again, we see this increase. But what's perhaps more interesting is this is not a linear enhancement of the mechanics. What we've shown is we only need to populate or occupy 20% of the protein to get this enhancement of mechanics. So that's a scaffold where we have 20% enhancement allows you to see this dramatic change in mechanics of the system. And we really wouldn't have known that beforehand. Another striking difference between these systems is that because proteins unfold under force, the network that forms is incredibly different. So in these examples here, we can remove nanostaples or covalent bonds, disulfide bonds in the protein to trigger more protein unfolding, to make it more force labile. And the structure that results is really very different. And what we've done more recently is started to manipulate that so we can favor either unfolding or entanglement to change the material properties of these systems. So if Giles could hold up some fingers to represent how much time I have left, that would be really useful. Okay, brilliant. Okay, so these are the challenges that we've been working towards, and in my final 3 minutes, I would like to tell you the work that has really inspired and some of the new directions that we're going in. And of course, I'll do that very briefly to stick to time. We're really interested in structure and connecting structure at different length scales. And in a really recent project, what we've done is combined wide and small angle neutron scattering, and that has allowed us to look at the hydration and the assembly of a mini protein, a beta hairpin. So what you can see here probably looks like something you've seen with molecular dynamics simulations. This is actually data that's come from a neutron experiment. So it's really powerful in terms of the rich information we can gain. And we would love to talk to other people in the Bragg Centre. Perhaps you have a nanoscale building block and you want to know how it assembles in solution. And we think this could be a tool that could be applicable to other projects across the centre. So please come and talk to us. We're also interested in dynamics. So more recently, we've been working with the diamond light source to develop an in situ SACS technique. And this allows us to capture the evolution of the network structure in time. And you can see that represented here. So how is this network evolving, and how can we manipulate by changing the ability of the protein to unfold during network formation? There we go. So let me go back to this example of blood. So I said it's this beautiful system. Fibrin is a long and thin, sticky protein, and it's part of a much more complex hierarchical structure in biological systems. So these systems are beautiful, but can be quite difficult to dissect. So one of the things that postdoc Matt Hughes has been doing, he's been particularly focused on this question of the aspect ratio of the protein building block. In a lot of these fibrous networks, the aspect ratio is long. Is that important for network formation? And I would encourage you to visit Matt's poster later on today, where he'll present some of his most recent work, where we've used protein engineering to manipulate the aspect ratio of the building block and shown just how important that is for the network formation, mechanics, and structure. So we're working towards applications with these systems too. So just last year, a PhD student, Chris de Bruin, who is now in Boulder, Colorado, she was embedding microbubbles into these protein networks and using ultrasound to trigger the rupture of the microbubble towards triggered drug release. And what's really interesting is what happens to the network around that microbubble in the presence of this huge micrometer-sized object around which the network is trying to form. We're also trying to really exploit the mesoscale network of these protein systems. So I said we've got these heterogeneous networks where we have clusters of folded proteins, and we've got this intercluster region. So the porosity of these systems is tunable, and by understanding how the network forms, we can tune that porosity towards manipulation of small molecule diffusion and release. And so you can see that in this little cartoon that Ahmed has made. And if you'd like to learn more about that, again, I'm going to signpost you to Ahmed's poster later on today. And then related to that, we use scattering techniques to get information on the mesoscale structure of our systems, and we rely on models to extract that information from reciprocal space. What would be lovely is if we could connect that to microscopy techniques that allow us to monitor the network in real space so that we can begin to develop a better understanding of the porosity of these systems, which has been demonstrated before, but perhaps more challengingly, we want to start to identify the clustering and this heterogeneous structure that we've identified in scattering. This is incredibly challenging, and I'd love you to visit Dimitra's poster, and she'll talk about the great progress that she's made working with the LIMAS facility. So I'm going to finish up. We're getting started with this ERC-funded project, and I'm not going to say too much about this today, but I'd like you to think about this as a Lego bucket with all of your favorite pieces of Lego. And I'm sure if you think about that, there was maybe one piece of Lego that did something really cool, and when you were building something, you always wanted to have that cool bit of Lego in whatever you're making. And that's what we want to do. We want to really start to have fun with the functionality of some of these protein building blocks. They are our scaffold, and we want to understand how we can tap into their responsiveness to get these materials to do some really exciting jobs. We're not quite ready to share that yet, but we'd love to talk to you about it if you've got ideas for things that we can do, and we can certainly keep you up to date with the progress that we've made. I'm going to finish. I'd like to acknowledge a really, really talented interdisciplinary research group, and I've signposted to those that are presenting later today. We're very happy to be members of the Bragg Centre because we are motivated and inspired by the other materials research that's happening here, and we're benefiting hugely from the infrastructure that is linked to the Bragg Centre. We've also loved having the summer research students through Royce and Bragg. I'd like to acknowledge our collaborators across the university, and many of them are Bragg Centre members, and they're in the audience, as well as our collaborators at the ISIS and Diamond facilities. Thank you all for your time.
Thanks so much, Lorna. Do we have any questions? We have a little bit of time just here.
So in this building job, compared to synthetic polymers, can you say anything distinctive advantages when you chose proteins as building block?
So I guess these are synthetic proteins in the sense that we're engineering them in the lab, right? So we're using protein engineering to make polyproteins, so that in that sense they're synthetic too. I think for me the main advantage is the one I alluded to in the last slide, which is this diverse functionality. So we continue to learn more about how powerful proteins are with techniques like high-speed AFM. We can understand their dynamics and how that's related to their function. And that's the kind of rich functionality that we want to tap into with these materials. Yeah.
Thank you.
And to Lorenzo.
Great talk, thanks. You showed towards the beginning this kind of heterogeneous structure in your protein hydrogels with like the more dense parts connected by the unfolded proteins. Yeah. So why is that the case? Why some proteins unfold and some don't?
Yeah, that's a brilliant question. So we think it's related to the way that force is propagated through these systems, and of course that's extremely difficult to model at the minute. And we would love to talk to people that are able to do that. So what has been demonstrated, that paper that I mentioned, the Buehler paper, so they're looking at PEG-based networks, and they've developed a coarse-grained modeling approach that allows you to look at force propagation through these percolating networks. And what's really interesting is you have these hotspots, you have these areas where the force is higher. And I think in those areas, it's more likely that these proteins are going to unfold. So what we've learned from the single molecule community is that the stability of a protein is really connected to the way the force is applied through the system. Pull it this way and it unravels quickly, pull it this way and it's mechanically robust. So we need to understand how these proteins are connected in the system, what force each protein is experiencing, and that will help us understand how these networks are forming in terms of the heterogeneity that you mentioned. Yeah.
I think we'd better move on, and Ashley's already storming the stage. So let's thank Lorna one more time. Thank you very much.
Yeah, okay.
Good. So our final speaker today is Ashley Victoria. She's in the final stages of her PhD here at the University of Leeds in the School of Chemical and Process Engineering. And we're going to hear about some of her work on optimization of all cellulose composites.
Thank you. Can you hear me okay? Yep. All right, cool. Yep, my name's Ashley. I'm going to try and cram the last 2 years of my research into 8 minutes, so wish me luck. So we get there. So I've been working on all cellulose composites. So for anyone who doesn't know what they are single polymer composites where both the matrix and the reinforcement component are made entirely of the same material, cellulose. Cellulose is a naturally abundant biopolymer with inherently good properties. You're probably wearing cellulose now, I'd say most of you, because it's also found in textiles as well, in the form of cotton, flax, and also regenerated cellulose such as lyocell, tencel, that kind of thing. You can produce ACCs using a partial dissolution process rather than melting. So in a suitable solvent, you can partially dissolve the outer core of a cellulosic fibre and leave the inner core undissolved, and that can serve as its reinforcement and provide the strength, and the matrix can kind of hold it all together, and so you get a strong interface there. So they do have a potential to be a sustainable alternative to your fossil fuel derived materials. So we're quite interested in what these materials can do. When it comes to partial dissolution, one of the challenges is making sure you get a good balance between the matrix and the reinforcement, because you want to get enough strength, so you want to limit that fibre dissolution, but you also need enough matrix to hold it all together. So to kind of overcome this, we've been using interleaved cellulosic films as an additional component to add in the processing of these composites in order to achieve bonding between the fibres, but also between the layers. And so some work that published last year was just kind of like introducing this process where we use this interleaved cellulosic film and we put it where it's needed in between cellulosic layers so that you can get good interlaminar bonding between the textile layers. And over here, what we found was that we could still partially dissolve the fibres, but also fully dissolve that film so that it not only sat in between the textile layers, but it also infiltrated through the fibre assembly. So this is just a quick schematic of our process. What we do is we've been using ionic liquids, which I won't go into too much detail in because I've got 8 minutes, but we use ionic liquids. We impregnate a textile and a cellulosic film stack. We then process it under applied heat and pressure, coagulate that, add it to water overnight so that the solvent can be removed and the cellulosic bonds can form again. And then we dry it and we form our solid cellulose composite. And so what we found was that with a good configuration of solvent with some co-solvent in order to allow us to process it at room temperature, we found that we could get partially dissolved fibres, and as I said, we could get that full dissolution of the film. And we get an increase in Young's modulus. The middle graph is peel strength, which is literally a measure of that interlayer strength, which was huge. And we only had to sacrifice a little bit of that tensile strength. So there was some interesting work there we thought we would take forward. What I want to talk to you mostly about now is actually our use of statistical design of experiments, which is just some work that we've been doing recently. So in the previous work, I kept my temperature, time, and my pressure under processing fixed. And the thing that I looked at was this kind of one-factor approach look at my solvent configuration, how much ionic liquid and how much co-solvent I'd have, and overall how much solvent to my cellulose I'd be using. And so this is what you see here with a little cube here, this one factor at a time. And you sort of see that you've got this kind of design domain where you might be missing what's going on here, where there might be something even better going on. So something like design of experiments, you have that design domain and you've got carefully designed experimental runs that allow you to do a bit of statistical analysis and start to predict and infer what might be happening, and maybe there's some sort of better region around there. So in this work that got published just before Christmas, I looked at the dissolution, time, temperature and pressure, and those were my kind of factors that I varied. I had a series of designed runs, including some replicate center points, so we had an idea as to whether we could actually replicate this process if it wasn't completely random. And basically, with a full factorial, it's a nice good screening approach to give us an idea as to what's going on in that kind of design space, if we can keep good quality. And in this design, our points are just upper and lower limits of our factors. And so just very quickly, I'm going to go again. it's limited for time, but we looked at Young's modulus as a key response, and just doing some statistical analysis, looking at the significance of these factors within a confidence level of 95%, and looking at various factors when we did this analysis, and looking at our coefficient of variance, whether we could get a relationship between Young's modulus and these factors, and whether it was applied to the data quite strongly, and you know, kind of see how well it could actually describe that data and capture that relationship. And so we found that actually like pressure wasn't significant, but time and temperature was, and there was this kind of like linear relationship potentially going on. But if you look at these center points over here, these were looking quite consolidated and very good, strong composites compared to the rest of the full factorial runs, and actually the properties were quite good. And so you see on this ANOVA analysis here, this particular significance here is a curvature, and basically in the analysis, in this kind of like least squares fitting, it's kind of picking up the idea that actually the center points were pretty good, and so it might not be appropriate to just capture it just on a linear scale. So just very quickly looking again at the characterization of the upper and the lower limits. So like one of the composites made at the lower time and temperature, and then the composite made at the higher time and temperature. You can see there's that major difference in that consolidation and also the mechanical properties, but you do see there's a little bit of degradation here as well, which is kind of another sort of factor to think about. And again, just like reminding us of these center points that had that good had a nice balance of consolidation, but also our composites were not damaged as well. So the next stage of that was something that we call response surface methodology, and the idea was that I wanted to just explore looking at the process of design of experiments and going through the phases and see if it actually is useful to this process. And with response surface methodology, we can allow quadratic effects to be captured because we've got more runs. And so going back to our little cube, we've actually got some extra points here now that will give us a little bit more information, so we might be able to see if there's some quadratic effects. There's lots of different response surface methodologies. I'm sure some of you are bigger experts than I would be, but I chose an inscribed central composite where we can pick some nice runs within our design space to make sure that I don't kind of go too far out and start degrading the quality of my ACCs. Got some extra runs, and then looked at, again, a kind of least squares analysis with an extended model that would take into account quadratic effects. And we could capture Young's modulus reasonably well. And so where we had originally our linear relationship, we now could see that actually the significance of time and temperature were coming more from quadratic effects instead. We also got some nice information in all this analysis to kind of relate in density to the Young's modulus, and found there's a very strong correlation. And also just looking at this kind of like contour plot that's made from that model, which kind of see there's this sort of nice little hotspot here of where our Young's modulus is maximised. So using that information, the final stage was optimise, validate and characterise, just to see if actually this model can do anything. And so what we did was using Derrida's desirability function and using that relationship in the model, kind of identified a series of combination of time and temperature that would maximise our Young's modulus. In lab, I went and did those conditions and tested them, and then did some like statistical analysis just to see if actually what it was predicting was very similar. Our prediction was 3.4 GPA, and our actual samples were an average of around 3.3. And then finally, we did some characterization of just other KPIs, such as tensile strength, failure strain, appeal strength again, density, and all of these sort of characteristics were very favorable amongst all of the rest of the design of experiments run. So it was a really nice study. I didn't believe it myself. I did quite a few samples just to make sure it was actually not fake. And so this was a really nice kind of rounding off of saying, okay, we've actually used this. So very quickly, I've got a bit of time, just in conclusion, just exploring the statistical design of experiments, applying it to the ACC making process, finding that relationship between Young's modulus and processing factors. Prediction was identified there, which we then validated in lab, and these samples actually did exhibit a good balance of mechanical properties and physical properties. And so now my future work is just looking actually at going into regenerated cellulosic fibres. So this was using natural cellulose, which is cotton, something which we used cotton, but I'm now looking at regenerated cellulose, which is actually spun from dissolved cellulosic solution. So again, you might be wearing it. It's lyocell, and actually taking that forward now to see if we can think of other ways that we could take this forward to a commercial process. Very quickly, I just want to thank my supervisors, Peter and Kieran. Thanks to my Biopolymer Dream Team research group and my CDT, EPSRC, for funding this research. Thank you.
Great, thanks, Ashley. Yes, you did get through that in 8 minutes. Thank you. Do we have any quick questions for Ashley, please? Yes, Lorna.
Thank you, Ashley. Can you tell us a little bit more, in the design of experiments, what would be your ideal? You talked about an optimized sample. What are the ideal parameters? Is it Young's modulus or?
Young's modulus is a factor that we looked at specifically because it was, at the time, it was the one that we could capture the strongest. And so in the initial exploration, it was just sort of, can we actually use design of experiments? Does it capture the data? Is it too random and too noisy? And so we could capture Young's modulus very well. And it's a property that we would like to maximize. Tensile strength, when we've been making these composites, particularly as we've found, kind of sticking with that design space, it tends to be quite unreactive to the process, which is a really nice benefit of using the film, because actually it's not sort of like jumping around, whereas Young's modulus is still sensitive, and it would be nice to be able to push that Young's modulus up a little bit higher. It's not where we want it to be, and sort of like future work is looking into how we can push that, but we were finding that from our sort of the first work that I did, we've actually been able to increase that Young's modulus, and it was just, for me, it was more of just this fun exploration as can we actually use this technique to then reduce the amount of work that we're doing, but it is an interesting parameter for sure.
And I think I noticed you had an estimated void content. Do you have any plans to measure sort of porosity or void space?
So the way I did that was actually going old school, measuring several samples, taking the volume of that sample, weighing it, and then taking an average density. And then from that, to working from the average density of like amorphous cellulose 1 and cellulose 2 density, I sort of had an estimation of what that void content would be. It would be really nice to do more, and having the microscopy images certainly have given us a bit of an idea as to whether we can actually see the voids. But I know there is some work in our group that's looking into some new techniques to actually look at the 3D structure of the composites, and I think that would definitely be a next step to really see that and be able to actually measure it, like get some really nice images, use some imagery, measure it, and really get into that.
I think probably in view of time, and I notice in the programme it's drinks time now, we better move on. Let's thank Ashley and indeed all of the session speakers again. And I'll just hand back over to Andy to tell us what we're doing next.
Okay, thank you, Giles. And yes, thank you to all our speakers from today. All three sessions were fantastic. So indeed, it's time we head back over to the refectory now for some drinks. We have alcoholic and soft drinks available. There's lots of posters still to see. We have the RSC here to talk to, quantum design to talk to. And for this evening, we now have a VR clean room experience to have a go with. There are 3 VR headsets which are provided by the University of Swansea to explore basically the inside of a clean room. So really do have a go at that. So without further ado, I think I'll close day 1, and thank you to everyone online for joining.